Good morning, everybody, and welcome to this very special event where we have assembled to honor our very own Dr. Sharad Sharma. We all know Dr. Sharma as a very thorough professional at work and a very humble human being in his personal space. And he has been one of those elite group of scientists who actually dreamt of having GLP in India, particularly in the public funding institutions. He got trained in GLP way before NGCMA even came into being. And it was those initial discussions of his and his senior colleagues with Dr. Nigel Dent at OECD and Dr. Deborah Kioy at WHO that actually set the ball rolling of having GLP within public funded institutions as well in India. And his dream got wings when CDRI also took this up, and the GLP was established, and we got a GLP certification here quite some time ago, and Dr. Sharad Sharma was one of the torch bearers of that exercise to have this here. He has spent over 28 years in CDRI, and presently he's heading the Department of Toxicology and Experimental Medicine, and he has contributed immensely to multiple regulatory toxicology studies and the drug development program of the Institute. So we have assembled here to honor him today, and we'll be having this one-day symposium on GLP studies, requirement for IND submissions. And we are extremely thankful to our speakers today, Dr. Sebastian Joseph and Dr. Manthan Janodia, who have come here physically. We were to be joined by Dr. Ekta Kapoor as well, but she had some last-minute change in schedule, and she will be now delivering her talk online. We thank all the speakers for coming over here. And to begin with the proceedings of the event, may I request Dr. Radha Rangarajan, Director CSIR-CDRI, to welcome the guests.
Thank you, Amar. Good morning to all of you. It's a bittersweet moment today. We are, of course, here to honor Dr. Sharad Sharma, who is retiring at the end of this month, and so is within days of leaving an organization that he has been part of for 28 years. He has been one of the pillars of this institution. So we are honoring him with this symposium, but also in our hearts we know that he is moving away. It is sad that he will move on. Of course, there's the next innings in his life, but as an institution, he will be dearly missed. I want to welcome all of you to this symposium. It's a topic that is close to Dr. Sharad Sharma's heart. Of course, a very warm welcome to our speakers, Dr. Sebastian Joseph, Dr. Manthan Janodia, and our current and former community members, Dr. Madhu Dikshit, former director, Dr. D.K. Dikshit, Dr. R.K. Singh, Dr. D.S.R. Murthy, Dr. Kalpana Murthy, Dr. J.S. Srivastava. Thank you very much for being here and for participating in this special event for Dr. Sharad Sharma. As Amir said, Dr. Sharma was keenly interested in establishing GLP toxicity facility, and of course, Dr. Madhu Dikshit played a very, very, very important role in all of that. But together with Dr. Rath, I think we can say that this institution created a facility that is so very special and so very necessary for the kind of work that we do. And then the main, having a facility of this nature means constant management and constant training and a very high level of diligence in all that happens. Dr. Sharat Sharma has been exceptional in the way that the facility has been, the GLP systems have been managed and maintained in CDRI. You know, in any drug discovery and development process, the one thing that we can't go wrong with is the determination of safety margins, the determination of how safe a compound is for entry into clinical development in humans. So the buck stops with the person who signs off on those safety toxicity studies. In Dr. Sharma, I found that person whose integrity is exceptional, whose clinical judgment is exceptional, and whose understanding of toxicity is exceptional. And therefore, you know, when I turned to him and said, do you think this compound is safe? I knew I could trust his judgment. That was a very, very important and critical part of being able to take compounds. You know, it has played a huge role in our ability to move things forward or to determine the kind of studies that are needed in order to de-risk the compound and make a go, no-go decision. So for all of that, Dr. Sharma, I thank you from the bottom of my heart. You have really stood with this organization and done the right thing at every turn, and that has been of exceptional value to this institution. You know, Dr. Sharma has been, he's always had a very cheerful and you know, positive demeanor, extremely helpful, always willing to work on solutions. You know, there have been times when he would come to me and say, there's a problem, this is a problem, this has been the problem, etc., etc., and then and then he'd say, and this is the solution, and that always made it easier for me. And oftentimes he was fighting for things which didn't have a direct impact on him, but which benefited members of this institution, and he did it selflessly. He did it, you know, with the same enthusiasm and energy that he would have had he been directly impacted. You know, so his selflessness, his practical mindset, the clarity that he brought to problems, this has always been very, very positive for me in all of my interactions with him. And of course, Dr. Sharma, you've been a great mentor to many in this institution. You've been a friend to all of us, a very exceptional colleague, and so your legacy will live on. Today, you know, we want to talk about some of the topics that can be of learning value to members, to all of us who've gathered here, as well as perhaps give you assurance that we will continue to lead, we will continue in the path that you so firmly stood on. So with that, I will pass it back to Amir to take on, to continue with today's symposium. Thank you very much, Dr. Sharat Sharma, for all your contributions.
Thank you, ma'am. Moving on with the program, we'll have the first talk of the symposium delivered by Dr. Sebastian Joseph. Dr. Joseph is a veterinarian and a board-certified toxicologist and currently working as Senior Director at Cencora Pharmalex. He has over 18 years of industrial experience that includes drug discovery, non-clinical development of drugs, experimental toxicology and pharmacology, good laboratory practices, computational toxicology, human health risk assessment, environmental risk assessment, and medical writing. Dr. Joseph is currently involved in strategic development and consulting services in pharmaceutical product development with special reference to non-clinical and toxicological requirements, non-clinical evaluation strategy in support of IND and NDA, risk assessment of pharmaceutical active ingredients and excipients, health-based exposure limit derivation, impurity qualification and of APIs and drug products, and environmental risk assessment of pharmaceuticals. Today, Dr. Joseph will talk to us about IND enabling preclinical studies, scientific and regulatory considerations to ensure subject safety. Dr. Joseph, please.
Thank you very much for the introduction. Good morning to you all. First of all, I thank CDRI, Dr. Radha, for inviting me and to be with you today. I consider this as an opportunity for me also to learn from you. So thank you very much. As Amir mentioned, so I will be talking a bit about the regulatory and scientific consideration when you are preparing for the IND. For any pharmaceutical, when you are about to test, about to give it to a human being, I think there are three most important aspects. So there are no formal slides. I could not complete our internal review process so on time, and therefore there will not be formal slides, but I will try to in other ways I try to cover and make it up, and I consider this as an opportunity because that way I'll be able to hear from you as well. Instead of you listening to me alone, I can listen to you as well. So there are three things: efficacy, quality, and safety. So these are the three aspects that we frequently talk about when it comes to a pharmaceutical. And you all know that what is the most important. Drug has to work, certainly. It should cure the disease, but it's very important that it is not harming the patients or subjects. And this is more important for when you are going for first-in-human trials, because in most cases you will be testing this in the healthy volunteers, although some studies you may test in patients as well. So when you are about to go for IND, the most fundamental question that comes up is, what studies I need to perform or what data I need to generate? Now, in my initial days of a career, what I used to do is, I used to take out regulatory guidelines and look at the list: acute toxicity, repeated dose toxicity, genotoxicity, and so on. But with time, I realized that there are better way of doing that. So today what I do is, when there is a question, what studies, I assume that I am going to participate in that trial tomorrow. And if I am going to receive that investigational compound, what all things that I need to have, or what all data that I need to generate, so that I am convinced, first of all, that the investigational product that I am taking is safe to me. Now, that is trial participant. So when we talk about IND, safety studies, trial participant, I remember a conversation, an incident that happened to my brother. My brother has undergone cardiac surgery, stent implantation. He's having 3 stents in his heart. He's taking continuous medication. And one fine day, his consulting doctor asked, there is a new compound that we are testing. A company has developed and we want to test. Are you okay to be a participant? And doctor said, you need not to answer. You can go back and next week we will again call you. And doctor provided some handouts. He came home and then called me and asked, hey, doctor told this, can I take that particular medicine? I said, I need to study about it. Then he gave me the literature, I studied, and then afterwards I told him, you can participate in the trial. Then he asked me, Are you sure? What will happen to me? I am already a cardiac patient. My heart is not working properly. My BP is not normal. I am taking cholesterol lowering drug. I am having hyperlipidemia. So is it really safe? By taking this, whether my kidney will get affected, my heart will get affected. I am already having muscle pain, back pain because I am taking statin. And so on. He started asking so many questions. Then I told him that you can go ahead, there's no problem. Then he asked me, his son was also sitting with me and he was listening. He asked me, on what basis, uncle, you are saying that? Then I told that, I showed the data and said, this compound was tested in animals and there were no major concerns. Kidney, liver, animals' behavior, everything was okay. There were some liver enzyme elevation, but that happened only at very high dose levels. Uncle, are you sure? You are talking about that based on some data you generated in rats. How sure that this is going to be safe, my father? Then I told, no, this is not only in rat. Even this compound was tested in dog. And they tested at very high dose levels, at several fold higher than what your father is going to receive. Then he was a bit okay, convinced. Then finally, he asked one more question. This is all generated by pharma companies. How I can believe? How sure that they indeed performed these studies in a proper manner? And the data that you are talking, are they reliable? Then I told that these studies are not simply done, just like that. There is a system of good laboratory practice. There are regulations. There are stipulated norms under which these studies have to be performed. So they have performed under all these conditions, and therefore I believe this should be okay. But still there was some doubt. Still, are you sure, uncle? The problem was that they were thinking that this is all done in animals. How safe that it will be in human? Then I told that it was tested, found to be safe at some dose levels in animals, both the species. And then what they are going to give to your father is much, much less dose than what was found safe in the animals. So with that answer, they got convinced both and agreed, and he is participating in the trial. So the point is that it's very important that when we plan for IND enabling studies, it's important that we convince the stakeholders. And the most important stakeholder is the trial participant. The next stakeholder, the regulators, the investigators involved. So we need to convince them in terms of the safety of the investigational product. Now, when we talk about safety of an investigational product, let's look at certain fundamental aspects. Can I have marker? So safety, is it working? Am I audible? Not much. So safety issues can arise because of pharmacology or because of toxicology. Safety issues can arise. Am I audible? Is it okay? No? So safety issues can arise because of pharmacology or toxicology. And when you look at the pharmacology issues may come because of the intended target, on target, or because of unintended target, off target. So these on target on the intended target, off target on the unintended target, or on target at a different location. Your compound is supposed to act on the gastrointestinal tract on the 5-hydroxytryptamine receptor, but it is getting into the brain and acting in the brain 5-hydroxytryptamine level. So that's on target but in a different place, or it could be off target. Your compound is supposed to act on the 5-hydroxytryptamine receptor, but it is also acting on the dopamine receptor. So on target and off target, when the compound acts, it can bring about certain pharmacological effect which may not be desirable, which may compromise the physiology of the trial participant. So when that happens, we call safety pharmacology. When a compound affects the physiology, it will lead to safety pharmacology issues. Physiology essentially on any system in the body, but if the physiology is disturbed with respect to the critical system in the body, let's say for example cardiovascular system, central nervous system, or respiratory system, it can even endanger the life of the participants. And that's why it is very important that you need to look at whether your compound, investigational compound, is altering the physiology, the critical physiological system, 3 systems. That's how ICH S7 guideline came into force, where the guidelines stipulate certain set of safety pharmacology studies, of which the core battery studies are cardiovascular, central nervous system, and respiratory safety pharmacology studies. That's about the safety pharmacology. Here essentially functional change because of the receptor or drug target interaction, functional change. So there can be certain structural change also because you are exposing your trial participants to the compound. So those structural change, this can result in toxicological effect. Now it's very difficult to differentiate between pharmacology and toxicology, but just to ease my job here, I am trying to classify these as a different set of effect, but in reality this both happens together. So toxicology, what we do, in addition to functional change, we also look at structural change. So that is what we study in toxicology. Now broadly, there are 2 types of toxicological studies. One is general toxicology study, and second is special toxicology study. Now if we go back to questions from my brother, he was asking whether the compound acts on my kidney, liver, muscle, and so on. He was worried. So all those things we study under general toxicology study. So it's general because we study everything in that kind of a toxicology studies. In those studies, what we study from head till toe, every system, every organ, every cell in the body. That's what we study under general toxicology studies. And therefore these kind of studies are the very critical studies, very, very important studies that regulators look at, regulators review very keenly while reviewing the IND dossier. So here there are requirements. I am not going into the details of regulatory requirements, these are all there in the guidelines, but I try to kind of share my certain experience and certain tips. So the current requirement is that you will have to study in 2 different species, and the study should be of duration that is at least equivalent to the duration that you are going to test in the clinical trial. So what we do here, we give the compound and evaluate every organ system in the body. So coming to the special toxicology studies, now you want to involve women of childbearing potential. So then you need to evaluate whether the compound is going to be safe for that kind of a target participants. So then you will have to perform studies that specifically evaluate effect on fertility, effect on female reproduction, effect on the growing babies, pregnancy, duration of pregnancy, and overall development of the child. So they are all studied under reproductive and development toxicology studies. So usually, as you know, in the first in human trials or initial phases of clinical trial, women of childbearing potential are either not included, or if they are included, there will be strict pregnancy prevention measures. So therefore, these kind of reproductive or developmental toxicology studies are generally not done. So another kind of studies is the genotoxicity studies. So when you perform a general toxicology studies, it is you are evaluating genotoxicity indirectly, but not really in a direct manner. Therefore, you design specific studies intended for evaluating the genotoxicity. You will do in vitro, in vivo. So what kind of studies? It's all there in the guideline, and I'm not repeating them here. Essentially, in vitro and in vivo studies are required. And the next kind of effect that can arise as a follow-up to the genotoxicity is carcinogenicity. Again, carcinogenicity studies or carcinogenicity as an effect is important or relevant when you are exposing your trial participants for a long duration of time, which typically happens when you are in phase 3. And therefore, again, carcinogenicity studies are not really required when you are going for IND or initial phases of clinical trial. Then there are other kind of studies, let's say phototoxicity studies. Now, especially that's important when you are going for a dermal preparation. You are testing a dermal preparation. Again, those studies may not be so important in the early phases of a clinical trial because you will put in certain risk minimization measures so that participants are not exposed to sunlight. Depending upon the phototoxic potential, you will take certain measures. But at times, these studies are also needed. And then another kind of studies that you often come across in the IND dossier is the local tolerance studies. Again, as I said, local tolerance is usually studied as part of general toxicology studies, and standalone general toxicology studies may not be needed in most cases. So what you have seen here, it's all the broader kind of studies that are required to evaluate the safety. Now, any questions so far? Any questions? Yes.
It is impossible to convince regulators and patients both about the safety. Are you agreed to this view?
Now, the type of studies that I mentioned probably not covering the entire thing. Is that your question? No.
Can we avoid the use of animals to establish preclinical toxicity?
At present, no. We don't know in future what's going to happen, maybe after a few decades. But I think currently, without animal testing, probably I may not give my consent to participate. My kith and kins also I may not allow to participate, and therefore I believe even the common public also may not say okay to test the medicine. So without proper testing in animals, at least today we are not in a position to ensure safety.
Probably because what we also feel when we are going through in ethics committees, and Dr. Dixit will share with you, when the GLP studies we go through it, and we look for whether the studies have been conducted in the whole animal and then in vitro. So I think whole animal study is very important. It must be. But that is what you are telling me.
Yes, yes. All animals, live animals.
Yes. Then you have to go for the specialist. Specialist.
The most important object of relative toxicology is to determine how to calculate first human dose, which is the most important part of the beginning of a trial. But you can know it and know it, you can calculate from fully. First human dose is the most important for it. Almost certain in the genotoxicity, certain local toxicity tests, alternative models have been they are most times transmitted.
That is the best available option so far. There are no better option today available, and therefore we are going ahead with the currently available best option. That's to make use of rodents and non-rodents and generate data. But if tomorrow, with the advancement of science, if something else comes up, I'm sure the use of animals will be reduced or even completely dropped. But today, as of today, I think there are no better way to ensure safety before we proceed with the clinical trial. Now, these are the set of studies, data that we talked about. But then how is the safety is established? You generate data, that's fine, but how is the safety established? The safety is established by way of a process called toxicological risk assessment. Toxicological risk assessment. So in this process, you try to identify risk, and you will also recommend risk minimization measures. So let's look at certain basic fundamental concepts. Now, in toxicology, all you know that fundamental is dose makes poison. So every chemical is assumed to be toxic. The only thing is that when the exposure is sufficiently high, even water is considered toxic, from a pure toxicology perspective. So therefore, this is a fundamental story actually. Everything is poisonous, only the dose determines. Now from that context, if you summarize this concept, I can say that ability of a chemical to exhibit its toxic effect, its unwanted effect, depends upon the exposure or the dose. So that is why we say risk is equal to hazard times the exposure. So there are two things actually in the risk assessment: hazard and the exposure. So hazard is the inherent property of a chemical to cause harm, inherent property. Now you may wonder, or you would have probably discussed also, for some compounds, compound is efficacious at 1 milligram per kg. The toxicologist wants to test that compound at 1000 milligram per kg. So you may wonder, or ask questions, why so high? We usually talk about 10x, 20x, 30x margin. But our toxicologist is saying you evaluate at 1000x, 1000 milligram per kg. Any thoughts on that? Why such a high dose? Why not stop at target organs? You may okay. You want to find target organs. You cannot find at low doses. Okay. This is a problem when the compound is safe, or relatively safe. In its toxic potential is less, you will face this problem. So you test at 100 milligram, 100x, there are no toxicity, 500x, no toxicity, and then you will go still high, 1000x, you may or may not get toxicity. Then the idea is, why you are going so high is simply to try to understand the hazard, simply to try to understand what is the inherent property of that chemical, inherent property. So when you know that inherently it is toxic, then you are comfortable working with that. So you have two snakes. One is the common rat snake, another one is cobra. We all know that cobra bite can even kill. On the other hand, even the rat snake, if it bites, nothing will happen. Now, if you are playing with these two snakes, okay, you will be very careful, right, with the cobra, because cobra bite, what will happen? So even while working with the investigational compound also, we may not really give that high dose, but in case if something goes wrong, it can happen anywhere, even maybe unintentional exposure, something dosing error, or anything can happen. And even if that happens, what will happen to the trial participant? You will know if you have really understood the hazard. Hazard is not risk, hazard will become a risk only if there is a sufficient exposure. So if a cobra is there and it is contained in a container and there is no way that cobra can come out and bite, there's no risk. Although cobra is highly hazardous, cobra is highly hazardous, but If as long as cobra cannot bite, there is no risk actually. The same way in toxicology also, our intention is to evaluate at very high dose, try to understand what this compound may cause, even if the exposure is very high. This is why we test at very high dose levels. And so therefore the entire toxicological, the safety establishment starts with hazard identification. The studies that we talked about, they are all simply studies to identify hazards. Once these studies are done, the next step is risk assessment. That is where we look at what those effects were produced, and that's where the NOAL concept come in. And how it is typically done, we all know. So there is already a guideline, from the US FDA. There is one guideline on calculation of starting dose for human clinical trial, first in human trials. So what you typically do, you look at the NOAL doses from all the studies that we talked about, take the most appropriate and relevant NOAL value, convert that into human equivalent dose, because animal dose and human, and because of the body surface area difference, they may not scale proportionately, so there is a conversion. And then you will get a dose once you convert that NOAL. And then in order to ensure that what you have, there may be certain deficiencies with respect to the studies that you perform, with respect to the precision of the NOAL that you derive. So there can be uncertainties. To overcome that, or to take care of those uncertainties, you apply certain safety factor. The default safety factor considered is 10x, and then you will get a dose. So that can be the starting dose for the clinical trial. So here, this process of calculation, deriving, this is the risk assessment. And depending upon the hazard and risk are separate, exactly. So just to give one more example, cyanide, it's highly hazardous, but the risk is low. On the other hand, pesticide, organophosphorus pesticide for example, it's compared to cyanide, it's less toxic, less hazardous, but risk is high, because farmers use pesticides for everything, fruits, vegetables, everything, and so that when you eat, when you live normally, every day you are getting exposed to pesticide. The exposure is more with pesticides, whereas cyanide is highly controlled, it's not available everywhere, very restricted, so therefore the risk is low. So that's the difference. That is exactly the fundamental actually. This all is fundamental to, while designing the studies, while making a risk assessment. I think these concepts every day it will be applied, and that's how studies are designed. That's how dose levels are estimated for the phase 1 clinical trial. Very basic concept. So people don't talk about these things because it's just default. It comes, but essentially the entire exercise is based on this concept of risk assessment. Risk is hazard times the exposure.
Animal pharmacology, and we are doing pharmacokinetic, and we found that 100 milligram is maximum bioavailability. Now we take 5 times more, 500 milligram. What is the use? Because these drugs are not being absorbed, and we say we are going to 2 grams. So what is the use of that range which is not being absorbed? So I think it was given in the ICS guideline. It is more practical approach, because here we are using a drug, not a pesticide, not a thing. We know a definite dose which is to be used. So based on the bioavailability, maximum bioavailability 5 times is safe range. Don't go much beyond. That's the one school of thought. But it's still 2 grams, and this is the part of NTCT and acute appendix also. So there are 2 schools of thought.
I think
I
that.
think second way is more practical. Unless we load on chemistry. Unless we load on chemistry.
So
It is not being absorbed, what do you find?
I think.
LD50 cannot calculate in the toxicity is an outward phenomena, but not an object purpose.
So I think it's a very important point that he raised. We talk about high dose. When let's talk about the toxicology study, general toxicity study, little bit study aspect. So we talked about the background, why this kind of study is important. Then we talked about the concept of hazard and the risk assessment. Let's see a little bit of study nitty-gritties of studies. I think that's what I understand that many of you are interested about. So also we will touch on the point that we raised. So if when selecting a dose level in a toxicology study, there are a few criteria. The first criteria is the highest dose should produce evident clear toxicity. This is our target one, clear evident toxicity, ideally no death. This is the first criteria. The next criteria is if a compound is safe, you will not get that toxicity. Second criteria is maximum feasible dose. Because of certain nature of the investigational compound, you may not be able to administer high dose levels, or you may not be able to formulate it properly to give a very high dose. So your ability to take higher dose are limited. Maximum feasible dose, that's another criteria. And the third criteria is limit toxicity, limit dose. How high you will go. Okay. Therefore, there are certain stipulated dose levels. For example, 1000 milligram is considered a limit dose for a repeated dose general toxicity studies, 1000 milligram per kg. Now here I think very important that when you are actually exposure, plot the dose versus exposure. So if the saturation is happening here, which is just 100 milligram itself, then there is no point in going 1000. That's the point that the bioavailability. So if you say I have gone up to the limit dose and you conclude that my compound is safe up to a dose of 1000 milligram, that's a wrong interpretation. I can conclude that my compound is safe up to a saturable dose of 100 milligram, exposure saturated dose of 100 milligram. That's the correct conclusion. So therefore, it is important to understand that relationship between dose and the exposure, and that's the reason why you will do initially pharmacokinetic and then followed by toxicokinetic study before you get into the actual toxicology study. So that even this curve also should be kept in mind while selecting the dose levels for the toxicity studies. And for safe compound, again, it is a problem. It may not saturate up to 1000. There may be a proportional increase, yet you are not seeing any toxicity. Then is it acceptable to cap it at 1000 milligram? In most cases, it is acceptable, but there may be situations where you target to administer to the trial participant itself very high amounts because of the nature of the disease, or you are working with such a target that you are giving very high, then you have to further increase actually. So this is default is 1000, but sometimes you may have to go still higher, or you will have to rework on the formulation so that you achieve higher exposures at the same dose or lower dose levels. So this may call for, if the saturation is happening at very low dose itself, that calls for reworking on the formulation so that you try different excipients, come up with a more different better formulation so that exposure happens, much higher exposure you will achieve in the when you test at these dose levels. So this is a general guideline about dose selection, and this applies to every study, not only general toxicology study. This guideline applies to even the reproductive toxicology study, embryo-fetal toxicity, carcinogenicity study, and everything. And another rule is there, how high to go, in terms of the area under the concentration. Typically, a default number of 50x. If when you are about to go for a first in human, you do not know what is the efficacious AUC. But you can make certain estimations and project using certain models. You will be able to project what's a projected or estimated exposure at the efficacious dose. So you can take that number as a reference and then try to achieve at least 50x at the high dose level if it is a general toxicology studies, 50x. So that's again another number to remember. This all we should keep in mind while working, but you may not be really able to achieve 50x, you may not be able to achieve 1000x. It's all fine as long as you have kept in mind all these points while designing the studies, and you bring in all these points in your dossier, I think then it will be acceptable to the regulators. It's important that we present our story. We did these things. There was no exposure. We reworked on the formulation. Then we got higher exposure. Then we did this study, but there was no toxicity. Then we did another study up to this dose. Then still there was no toxicity. So then regulator will okay it. There is no problem because you have systematically evaluated the hazard. The risk assessment, your risk assessment is based on a strong science. So if you go by this principle of risk assessment, risk equal to hazard times the exposure, the science, use of that science everywhere in your design, I think there will not be any questions from the regulators. So again, talking about general toxicity studies, or for that matter, any toxicity studies, I think it's important that we keep the target patients or target participants in mind. If we are testing for an indication that's only meant for men, you are going to use men as a participant. There is no need to include females in your toxicology studies, vice versa also. So although the guideline says male, female, 3 dose levels, but if you have a strong reason to say that okay, one gender is sufficient, then there is no need to perform studies in the another. Another question is duration of the toxicology study. How long? Repeated dose studies, how long should be performed? 2 weeks, 4 weeks, 3 months? So it's a simple answer is it should be, the duration should be at least similar to the proposed clinical trial duration. So that is a little bit about the design aspect, the dose selections. Any questions? Any thoughts?
Yes. In this case, suppose.
Yeah. I think even without mic, I think we are able to hear.
Okay. So as you said that for certain drugs, if it is positioned for men, no need to use female animals for the toxicity studies. I'll give one example. What would be the situation in this case? Phosphodiesterase 5 inhibitors, sildenafil, vardenafil, commonly known as Viagra, is meant for men, but then the drug can be repurposed for pulmonary hypertension. And in this case, if female is the patient, in that case, do these drugs have to go through the entire animal toxicity studies in female as well?
If you have initially thought about men indication and then you performed only studies in men, then you will have to go back and do the study in female. It again depends on your strategy, your plan. If you have some thoughts that I should repurpose in future so that I will not repeat it later, so let me include both male and female in the first study itself. It's a choice that you make, but in terms of the data requirements, there is no need to perform for a male indication. Tomorrow when you are switching to a female indication, then they will ask where is the female data.
Which has overcame the Schedule Y. They do allow only study as per indication only one gender. If rule does not allow, so we can do for one gender.
Yeah, you see, there may be a certain departure with respect to certain countries and certain
India,
regulations.
We are talking India.
Yeah, so guidelines are just guidelines, and
it is no
they
rule.
by default they would have recommended male and female. However, I believe that if it is justified properly, even to the Indian regulators, with using the scientific concept.
Now it is rule. It is no more a guideline. It is now rule.
Yeah, so that may call for rethinking on our rules and regulations, and make it more science oriented. That is what I am scientific and regulatory consideration. That's why I coined the term scientific and in countries. Yeah, if such a rule is there, probably they expect that it's to be performed in both.
Yes.
However, especially this when I am here in this kind of institute, that's where I can put forth these kind of thoughts so that, the concept can be challenged, because
you are open to decision by the authority, receiving authority. They can decide. They may give, depending on how you have presented your story.
No, but Karan, the point is, they are not going to reduce the N. They will still say 4 males and 4 males. If there was any point in avoiding females to reduce the number of animals, that might have been accepted. But that will be only some that will depend on the story you have written.
yeah.
You make it, if it is only for one gender, suppose as contraceptive, female contraceptive, you don't require in males. No. That kind of study, huh? Number of animals will be one gender only. You can reduce the number of animals, but depending on the indication. So you won't be taking female contraceptive also.
I think that's where the science should, we should talk more science and base our arguments on good science. That's where, I said I'm not going by the list and then, identifying what studies to be done. Instead, I come the other way. Assume that I am a participant to ensure my safety, what all things I need to have, and justify scientifically. In countries where, regulations and science more advanced, especially in the pharma, it's much easy to tell them that, okay, guideline says this number of animals. However, I took this less number because of XYZ reason, and, it's perfectly in line, from a scientific perspective. They'll say, okay, it's a rationale that matters, and I have seen myself, lot of departure from the standard regulation, standard guidelines. Lot of them, and we were initially thinking whether this will accept, but we tried and accepted, because I think the amount of science that gets into that argument, that's important, and the most stringent regulatory agencies, in the world, they are also getting convinced if we are putting forth good science. So science-based arguments, science-based rational design of toxicology studies.
A pre-IND submission meeting can sometimes be very, very helpful, where you explain the rationale and the science to the regulator upfront before starting, or before you submit an IND or while still planning, to get agreement on what makes sense as a toxicity safety package, and then proceeding. I think that way there are fewer surprises. I mean, at least with other regulatory authorities, these become binding. I'm not sure how that works for India, although pre-IND meetings are now available. Any experience with that, Sebastian?
I have supported several pre-IND meeting and scientific advice meeting with the US FDA and scientific advice meeting with European authorities, but so far I have not worked with our local. That's why, sorry, I do not have that experience actually dealing with our local authorities, but at least with the FDA and EMA, that's a very common approach, that especially when your target, your molecule is slightly different than the conventional, you are working with certain indications that are not very typical, and it's very common that we approach agencies, take their concurrence before we implement the IND enabling studies. But I am not sure how this works with our local authorities. Okay, so another, I will touch upon certain design aspects, species selection. Time? Okay, how much time left? 5 more minutes. So I think I sought certain questions, feedback initially, and I got some points. So I will just quickly touch on that. One of the thing was species selection, dog versus monkey versus rodent. I am sure, I think many of you are aware, but I think a critical criteria is that target should be expressed in the species that we are going to select. target sequence and homology should be consistent and very closely to humans. Thirdly, target is distributed in the same manner as like human, that's, in terms of different organ systems in the body. Target expression and its physiology is more or less same like humans. The body handle the investigational compound, the animal body, handles it the same way like humans. So these are all the criteria based on which a species is selected, and I think the pharmacokinetic consideration is, I think, very, very important in terms of what enzyme system metabolize this compound, what metabolites are produced, what is the extent of metabolism in different organs, and different ratios of different metabolites. I think these are very critical criteria on which you select a species. So if there are gaps in any of these points, so you will have to make certain refinement in your study design, your dose selections, and many more actually, so that the regulators do not raise concerns on your studies. Species selection. Yeah, so next, test item related. I think there was some question on the test item. Many people think that the test items used for IND enabling toxicology studies has to be a GMP grade test item. I would like to say that that's not the requirement at all. The test item used in the GLP study, and it has to be a good quality, means it should not be too dirty. At the same time, I would say it should not be too pure, because if you are using a very pure test item, tomorrow when you work with the clinical batches, and if some impurity level goes higher than what you tested, then you may have to again come back and do certain additional toxicology studies to qualify the impurity. So therefore, you should use a reasonably good quality, reasonably pure material, maybe let's say 98% pure material, and the GLP expects that your test item is properly characterized. So characterized means, you should be clear what that test item is. It's a chromatogram, spectra, and you should be sure that this is the test item that indeed you wanted. It should not be something else. So, and then the second aspect is purity, that I talked about, purity, impurity profile, and the third one is stability. How long is stable? If a study is of a 4-week, the test item should be stable for at least 4 weeks of duration. So if these things are met, that test item is qualified to be used as a GLP test item. GLP versus non-GLP. So is it mandatory that studies have to be GLP? Again, there may be certain regulations that mandates these kind of a safety studies have to be compulsorily GLP compliant. But in my experience, when working with especially US FDA or EMA, there may be instances where you are submitting a study, a critical study, based on which you are deciding the fate of the dose to be selected, the escalation plan, and so on, a non-GLP study, and still got accepted without further questions. It is, it depends on how good that study was, how good the documentation was, whether the design of the study was properly done, a good science is there behind it, and due to some reasons, the GLP could not be, it could not be done in a GLP compliant manner. This is common with the new therapeutic modalities, maybe advanced therapy, medicinal product, cell and gene therapy. And so therefore, in those cases, there may not be laboratories who can perform these kind of toxicology studies in a GLP environment, and it's absolutely fine to perform under non-GLP, but there should be a proper documentation so that if they want to come and audit, there should be everything, everything should be traceable. So therefore, it is more than the GLP or non-GLP. I think it's important that the study is designed properly and documented properly. But in general, wherever possible, the regulators expect that the key studies are performed under GLP compliance. So another thing is customizing your studies. We talk about IND enabling studies. There are certain type of studies that by default. But I think it's very important that you look at the target participant and come back and customize. So for example, you are working on a disease for a pediatric indication. You cannot perform a study in an adult rat or an adult dog. So you need to look at what is the age of the pediatric population that in which you are going to test your medicine. In most cases, usually pediatric indications, first they would have tested in adult and already in use. But there may be rare diseases. I have worked on few rare such indications, rare indications, orphan designations, where that disease is there only in children actually. By the time they reach adulthood, it's likely that they will die. That kind of unmet need, orphan designation. So in those cases, you cannot perform a classical general toxicity study 4-week, and you will have to match the animal's age with that of the trial participant. For example, when the rat or mice is born, it's considered their preterm. It's equivalent to preterm neonate actually. By the time they reach 8th day, that's when day 8 of a rat is equal to day 1 of a newborn baby. And by the time they reach around 22 days, it's as good as the baby reaching 2 years. So therefore, if you want to test a product in a 2-year-old child, then you will have to start the dosing maybe around day 20 using very young ones, or even neonates. You'll have to start from day 8. So therefore, customization of a study is important. That's where the gender comes, age, the route. Route is again some very very important aspect. There may be situations where there are compelling reason that you cannot perform by a given route, by the clinical route, and therefore in animals you will use a different route to conduct studies. So that's where the customization is important. So essentially, when we are designing, when we prepare for an IND enabling toxicology studies, we need to keep the trial participants' interest in mind, and we need to assume that I myself is going to take this medicine, so that to ensure my safety, what all studies I need to be conducted. So that, I think that is the important thing. And then you think from a scientific angle, everything, keep science at the center of your study design, and design the studies and conduct the studies. So when we do in that manner, we will be in a very good position to face any questions from the regulators. You have to preempt the questions. You have to proactively think what FDA may ask, and accordingly, or whichever the agency, and accordingly be prepared in the study conduct stage itself, design stage itself, so that you are better prepared to answer any questions. So that then and when you do that kind of an evaluation, a systematic evaluation, and ready with the data, I'm sure that you can ensure the safety of the trial participants. The first stakeholder, our first stakeholder is trial participants. The second,
That is, I think, would be.
yeah, I think
better.
both goes usually usually
I agree with you.
both goes hand in hand.
I agree with you. Trial participants should be get. But first objective of a pharma company should be to satisfy the regulator.
Now that's because the regulations are not up to the standard. That's why this question comes. Otherwise, you cannot separate regulation from science. If you see any our international guidelines, ICH or anything, everything pakka science. You say n equal to 6. Why? There is an extensive set of data already studied. Maybe hundreds of toxicology studies, and then came to a conclusion that around 6 number of animals will be able to detect, let's say, 10% difference, statistically. And then they said n equal to 6 or n equal to 5. So every point in a guideline, is based on a good science, and that we will know when you get into a guideline and try to understand, and you refer the references given in that guidelines. You will see that huge amount of data was evaluated before arriving at the decision that this should be performed with this many number of animals, this many number of groups required. Therefore, regulations good regulations are built on a strong science. But there may be situations where regulations are just like that, somebody wrote it and it's not based on science. That's where both may not go hand in hand. And when we see such things, I think that's where we need to work. We need to give feedback. As science is not, this regulation, there is a, departure. Both are not going hand in hand. Regulations. Yeah. We need to work on improving this system. Our regulation should be updated periodically. Updated. Yes, guidelines should come, and so that we ensure our citizens, our participants are, safety is ensured, and at the same time, animals and resources are not wasted. I think there is a balance required between these two. So these are some of the thoughts. Okay, yeah. So sorry, I did not use a formal slide presentation. Hope I did not confuse you. And I'm happy to, if you have any questions, follow-up questions, I'm happy to discuss and deliberate. And thank you for providing me this opportunity. Dr. Radha, thank you very much. Very happy to meet you all, and first of all visit this prestigious institute. So thank you.
Thank you, Dr. Joseph, for that extremely interesting talk and a very lively interaction. So we are extremely blessed to have, stalwarts of regulatory toxicology. The gentleman who was discussing about NOEL and AUC has served on a very long time on the IND committee of CDSCO, and he's presently heading multiple ethics committees as well, Dr. Chandeshwar Nath, and where Dr. Jayesh Srivastava also heading multiple human ethics committees also, so that's why his queries. And so we are enlightened today with lots of queries. Thank you so much to everyone, and thanks to Dr. Joseph for that interesting talk. Moving on, we'll have our next talk delivered by ma'am to hai sir, ma'am to goes without saying, without saying. Obviously, yes, obviously. Thank you, ma'am, for your guidance as well. So Dr. Manthan D. Janodia will be delivering next talk. He's a PhD from Manipal Academy of Higher Education, which he completed in year 2009. Before that, he did his Master of Pharmacy in 2003 and Bachelor of Pharmacy in 2000. He's a professor in Department of Pharmaceutical Regulatory Affairs and Management, MCOPS, Manipal. And he also serves as coordinator of Intellectual Property and Technology Transfer Office, Directorate of Research, Manipal Academy of Higher Education, Manipal. He is a recipient of UGC Raman Postdoctoral Fellowship at Department of Pharmacy Administration, School of Pharmacy, University of Mississippi, USA. His research is well funded by research grants from ICSSR, DST, and NCT, MIS, and other funding agencies. He has authored an interesting book on basic concepts of intellectual property rights in 2014, which has been published by Manipal University Press. Today, he will talk to us about GLP studies and pharmaceutical regulatory requirements. Dr. Janodia, please.
Good morning to each one of you. Am I audible? Yeah, okay. Let me first of all thank this prestigious institute for having me here and share some of my thoughts on this occasion of a tribute to Dr. Sharma. And I would try to complete my talk in next 30 minutes. My talk is categorized into three pieces. Basically, I'll be talking about history of how GLP came into existence. And then I'll be talking about some data from NGCMA impact analysis report, which is important for us to understand that GLP studies not only advances science, but it also contributes in indirect ways to the overall economic sector of the country. And then I'll be talking about pharmaceutical industry and IND requirements. And lastly, I'll try to put all these three pieces together and make a sense of what I'm speaking, right? So basically, we all know that this is something that is affected and that is important in the field of pharmaceutical sector. And that's where I'll start my presentation. So this is something that I'm going to talk about in next 30 minutes or so. Not necessarily in the order though. As we all know, all these practices, what we refer to as good practices, whether it's good manufacturing, laboratory, or clinical practice, all emerges because of some of these malpractices that happen. And we are very well aware of the disasters that have happened in the past, which led to implementation of a lot of guidelines that we see today, including safety, efficacy, and quality that we are speaking about. And Dr. Joseph has done a fantastic job of setting the stage for me to skip some of the slides in my presentation so that I very well manage my time as well. So I'll just go back to this example of industrial biotest laboratory, which many of you are aware of, but some of you are not aware of, which laid the foundation of GLP guidelines. And this was by serendipity that was discovered, for those who do not know, because when the data was submitted, and industrial biotest laboratory, IBT as it is known, it's a very infamous case. And that case led to the establishment of GLP facilities or GLP standards across the world. Now what had happened is, for those who are unaware of this particular case, is that there was a data submitted by a company and a whistleblower told about some of the fraud practices that were happening with terms of data generation by this agency. And this agency was very well a reckoned force in terms of conducting clinical testing or animal pharmacology rather. And it so happened that in this laboratory, when there was an investigation carried out based on the information by whistleblower, they found that the data, there was a scientific misconduct, scientific fraud that was being done. The data that was generated was not based on real science. The animals that were used were not taken care of. The data that was submitted to the agency did not have the correct interpretation of data that was generated. Now, in this facility, when the investigator went, there was a faulty water system, because of which the animals that were placed in the cages for the experiment were actually, the faulty sprinkler system was releasing a mist over a period of time. And because of this, there was a 4-inch flooding on the floor of where the animals actually had been died and decayed. So of 100% of animals that were used in this laboratory, 80% of animals had decomposed. So investigators had to enter this laboratory with a rubber boot, which was flooded with 4 inches of water. Many of these animals that were decayed, decomposed, were replaced without having any documentation. And data that was generated by IBT was being generated for companies manufacturing pesticides, pharmaceuticals, cosmetic ingredients, animal feeds, etc. And this data was submitted to the regulatory agencies for approval of marketing of this drug. So this fraud happened in the year 1976. And the investigation by FDA continued for almost 3 months. It was a whistleblower who informed the US FDA that there is a malpractice that is happening, and IBT had conducted this data for a company called as Syntex. And when the US FDA investigator, whistleblower is somebody who tips off, yeah, the name is not revealed, sir. That's why they are called whistleblowers. The safety of whistleblower is important. If the name is known, he doesn't remain a whistleblower. So this is something that happened. Tuskegee study. This was basically a scientist working in one of the companies, and then journalists investigated, and there is a long list of, the entire set of events are already, yeah. In this case, it was not the journalist, sir. Journalist went, reported the case, but the whistleblower was a person working in the company, right? So that's the case, and that's what happened, and because of which US FDA investigated it for 3 months, and they found glaring discrepancies in terms of protocol submission, data generation, data submission, replacement of animals without any records. So all this led to FDA, and eventually the reason is that IBT was one of the largest facilities that was conducting studies for not only the private companies, but even for US government. So 30 to 40% of studies submitted for approval in the United States were conducted by IBT. And it was found that of all the data that had submitted, over a period of 10 years, they had conducted almost 22,000 studies, of which 30 to 40% of studies were found to be having malpractice and scientific misconduct. Same thing happened with GD-Searle. Searle was having a well-established marketed product called as Flagyl, which is basically metronidazole, an antibiotic. So the data based on which they had got approval for short-term marketing approval of Flagyl was accepted by FDA. But when Searle wanted this drug to be tested for chronic therapy for long-term studies, that's where FDA became suspicious, and then they investigated that for long-term use of metronidazole, it is a carcinogen, and which was not reported in the data that was generated by them. So that's where GD-Searle again was investigated for 3 months, and ironically, Searle only submitted the first draft of GLP document to the US FDA, which was fine-tuned and implemented in 1976. So as we know, the tragedies become the cornerstone of us to implement any good practice that we are talking of. So basically, if you look at the history, entire history, as far as pharmaceutical drugs is concerned, all the good practices have come out of tragedies that have occurred. In this case, tragedy did not happen with the humans, but it was animals who were sacrificed, who had to go through all this, because of which the investigations and all these good practices that we are talking of came into existence. And these are the articles. I've just taken the screenshots which are available of the reports by journalists who later investigated and reported in newspaper, which became the talk of the entire country, and US FDA becoming stringent in terms of what they want to implement as far as approvals for testing on animal pharmacology was concerned. Spurious invention in the lab. So these are all, and this laboratory, IBT, had all the people investigated and they were imprisoned. The case was filed against them, and the trial, the jury held that the kind of crime that they have committed amounts to almost 40 years of imprisonment for each one of them, with a $40,000 fine in 1976. So this were all the malpractices which led to what we are today in terms of good laboratory practices, which basically depends on the principles of reliability, repeatability, auditability, and recognizable, right? So basically what we are discussing, what discussion happened earlier, depends on the studies that we are doing. Now, whatever studies we want to do should be planned. Now, what species to use, how many animals to use, all depends on the protocols that we decide, whether it's a pharmaceutical industry or the investigators. The entire study depends on what is there in the protocol. So your protocol becomes a strong document, and when you go to the regulators with the protocol which you have established, and studies carried out based on the protocol, whether you want to involve male or a female or both species, will be guided by what is there in the protocol. So if protocol doesn't require or protocol doesn't mention a particular kind of species of animal, then regulators will not question for a particular indication if you have already identified in the protocol. So all these activities are important as far as GLP activities are concerned. So your GLP activities should be planned. That means the experiments, the investigation that you're going to carry should be planned. It should be performed as it is described. It should be recorded. Now we have seen that in earlier case, there was no record when the animals were replaced. The dead animals were replaced with the new animals on which there was no test carried out. And the data that was generated using his pathology studies were submitted to the government agency or the regulators for approval. So that's where you have to have all the records of, and any amendment in the protocol also should be recorded. If you're changing the species, if you're adding the species, if you're adding the number of animals, should be recorded reported, archived and monitored. Archival is important because again, all this data that was submitted in case of IBT were based on the archived data. So investigators are able to pull out all the data that was submitted to them and identify where the discrepancies were, right? So archival of this data equally is important for us to retrieve it and find any anomalies if they are reported at a later stage. Now, these are the fundamentals of GLP, which I will not be going into details of because many of us are aware of. The GLP is basically dependent on resources that you have, characterization of test items and systems, rules, results, and quality assurance where audit is important. So these are the fundamental principles where GLP is implemented. Now, coming to GLP regulations, one of the good things that happened, and thanks to Dr. Sharma and the team, I mean, before I speak about this, let me talk about his vision that he worked on GLP even before it was implemented. And that vision can have multi-pronged impact in the entire landscape of the science and regulations that we are talking of. So this, India became a partner to mutual acceptance of data for GLP. Now, why this is important is any experiment as a part of GLP is conducted in India is accepted in 30 OECD countries. Now, OECD countries are developed countries. What is the impact of this? The data that is generated is not supposed to be repeated. So the experiments that you are carrying out are accepted by other regulatory agencies. So it saves you money. It saves a lot of use of animals. I mean, you can reduce the number of animals used in the testing. You can avoid repeat studies, same studies being repeated on the animals. And that has led to a cost saving of almost 300 million euros for all the companies who have followed GLP facilities. And these are the data from NGCMA impact analysis report. So this is an advantage of a GLP facility. And that's why I say that GLP not only contributes to the science, it also contributes in many other indirect ways in terms of what is being contributed to the general economy of a country. This is a harmonization procedure where India GLP lab data is accepted by the OECD countries. Now, non-clinical studies are mandatory when you are talking of pharmaceuticals. I will not go into details of other sectors, but pharmaceuticals is one which is important and it's contributing to the economy of the country. So I'll be focusing on why it's important for pharmaceutical products. We all know that we as humans consume these pharmaceuticals directly and it impacts us. So having a safety assessed in the beginning itself is important, not only in terms of what happens to animals, but also to us. Nevertheless, no studies are foolproof. Even if you establish a toxicity study in animal models, because animals do not replicate completely the human system, there are bound to be errors, right? And we have examples of drugs being withdrawn from the market because of safety even after marketing approval. But nonetheless, what it does is, it gives you a certain level of confidence to go ahead and test these compounds in humans with minimal risk, right? So you are trying to minimize the risk by carrying out experiments on animals so that it becomes comparatively safer for humans to consume when it reaches the market, okay? No drug gives you guarantee of complete safety, but the error, the risk is minimized for the humans. Now coming to impact analysis of GLP. Now, we have seen that in India in 2004, when the GLP studies or GLP laboratories had just started to begin, we had only 4 GLP laboratories, certified facilities. And then in 2011, I think Dr. Kapoor is going to talk about this mutual acceptance of data. When it came to 2011, mutual acceptance of data was accepted from India. India became a part of that, OECD, right, for testing facilities. The number of GLP certified laboratories as of 2020 were 50. So this is, again, I'm talking of indirect effect of implementing good practices. From 4, it went to 50, and still it is less compared to the size that we have, the kind of studies that we do, the science that we have, and the regulations that are evolving. And this again was implemented with the help of Quality Council of India, because we all know that Quality Council of India works in order to ensure that quality is maintained as far as the studies are concerned. The Indian testing facilities, as of now, have carried out more than 60,000 studies, and these studies are not only from Indian companies. These studies have already been contracted by other companies outside of the Indian borders. So this 60,000 non-clinical safety studies by Indian TFs have generated a revenue of 7,800 crores, which is close to $1.3 billion. Now this again is contribution to Indian economy. So when you have GLP certified facilities, it also contributes to Indian economy by generating revenue for these testing facilities and making it robust in terms of meeting the ever-changing regulatory requirements. Of this studies, 40 to 50% were Indian sponsors, which means that more than 50 to 60% of foreign sponsors now have the faith in Indian testing facilities for the data that is generated, which essentially means that Indian data is valid. And it essentially means that we have a scientific temper, we have qualified personnel, we have facilities which adhere to the highest standards of requirements, and foreign countries which normally are considered to be stringent in terms of their regulations are also accepting that Indian regulations and Indian facilities adhere and meet up to the requirements that are established by foreign facilities. The reason we are focusing on pharmaceutical is because the GLP studies, the contribution of pharmaceutical is almost 70%. Of all the studies, pharmaceuticals account for a major chunk of the studies because the pharmaceutical companies are the major users of these facilities because they have a stake in the game of selling the products. There are about 12,808 studies done only in 1 year, where CROs contributed to 90% of studies and in-house testing facilities were only 8%. Now, what has happened is many of these testing facilities are contract research organizations who do testing facility for other manufacturers. But some of these pharmaceutical companies have their own in-house testing facility. Now, this gives them an advantage of speed and scalability. And that's where and many of these companies who are research oriented have their own in-house testing facilities so that they don't have to rely. The data that is generated by them is, again, robust because they know that this is important in terms of time at which they submit this data to the regulators. The speed is important for them to understand. So there are 12 in-house testing facilities, and most of these facilities which are created in-house by the pharmaceutical companies. And there are about 14 companies who are both CROs and in-house facility. In addition to, in order to use and optimize the resources, they are also offering services to other players who are interested to carry out this TFs. Now, if you look at this data and the GLP certified laboratories, the highest number of GLP certified laboratories in India are in Maharashtra, Gujarat, and Karnataka. Now, if you try to correlate this with the sector that is pharmaceutical, these three states account for the maximum number of pharmaceutical companies. So they are clusters in a way where pharmaceutical companies are established. So it perfectly makes sense to have this kind of GLP laboratories in this particular states where Maharashtra, Mumbai is a hub of pharma industry, Gujarat has a sizable number of pharma companies, and Karnataka has a sizable number of pharma companies. So this aligns or it syncs with the number of pharmaceutical companies and GLP certified laboratories. Not to say that other states do not have, but you have to just find a correlation in terms of why you have these three states almost contributing to 33% of all the facilities that we have. Now coming to business value that this testing facilities generate. By the way, this is from the data that is taken from NGCM impact analysis report. Before 2010, the business value of this testing facility, that is the business that they have generated, was approximately 300 crores, which rose to almost 1400 crores, which is 5 times after the establishment of GLP certified laboratories. So again, it brings in forex revenue and contributes to the overall economy of the country. Now, these facilities do not come without the investment, right? We know that there is a capital and operating expenditure that is required, and that's where a lot of people stay away from going for GLP facilities. But nonetheless, those who are interested, especially the pharmaceutical companies, have invested money, and whatever existing facilities that they had been upgraded to GLP level. So they had made an investment, a total of almost 1100 crores of investment was made in order to upgrade the existing facilities to make it to GLP standards. The bright side of this investment is that this investment can be recovered, because if you are a part of MID, if you are investing in making your GLP facilities on par with global standards, it also brings in revenue which helps you break even in a shorter period of time, so that whatever investment you have made is recovered. So if you look at the global sponsors, even I was surprised to see that almost 21 studies were being sponsored from the United States. And we know that United States leads in the pharmaceutical research and development. So China accounts for almost 15 studies, and then you have for the whole of, you have the whole of Europe also coming to Indian testing facilities, GLP certified testing facilities, which essentially means that these are the companies who come to Indian GLP facilities in order to get their data which they want to submit to multiple agencies, which are a part of MID. Right? So there are global sponsors which otherwise were missing earlier. Now coming to the new drugs. Now if we look at the data, the number of new drugs that are being approved by US FDA is constantly growing. It gives us an indication that these requirements of GLP are only going to increase over a period of time. Why this is going to increase is because there is a rebound interest in development of pharmaceuticals. Indian companies are also upping their skin in the game by at least taking first steps in terms of carrying out basic and translational research, in terms of developing new drugs. There are a couple of companies who are doing this. So if we know that these numbers are only going to go up, it means that as a regulation, as far as in India and many other regulatory agencies, it is required as a part of law that animal toxicology studies are carried out in GMP compliant facilities if the purpose of the data that is generated is for IND submission. Right? So which essentially means that companies who are interested in developing new drugs will have to come to these GLP certified facilities, and they will have to get approvals for IND from these regulators only based on data that is generated at GLP certified facility. Now, this is again a commercial aspect that one needs to understand. These studies are important because if you look at this phase 1, most of the studies fail in phase 1 because of safety. If we keep aside commercial viability, phase 1 is where the most of the drug fails. And this is where we are talking of GLP, because if you establish the toxicity properly by using appropriate methods and protocol based on the guidelines given in the document by regulatory agencies, probably you can identify the safety at early stage, and the chances of failure in phase 1 gets reduced. Right? So it all depends on how you design your experiments. Again, it has to be documented properly in a protocol, follow. As Madam was mentioning, today we have the opportunity to have a pre-IND meeting with Indian regulator, which earlier was not available to my understanding. On the contrary, in the United States, US FDA gives this opportunity to sponsor to have meeting for pre-IND, pre-phase 1 submission, phase 2, before phase 2, and phase 3 trials. So that whatever data, whatever discussions, whatever apprehensions regulators have can be addressed. And that's why the failures at phase 2 and phase 3 over the years have reduced, because the US FDA offers this opportunity to the sponsors to come and have a discussion with them, which was much required in India as well, which is a positive sign. Now this is the drug discovery for those who do not understand why this is a high stake game. A drug discovery is not easy today, and pharmaceutical companies invest billions of dollars. So the chances of failure needs to reduce. Now, approximately 10,000 drugs are screened, of which only one goes to the market. And this entire activity takes almost 10 to 12 years, because you have to follow all the requirements, you have to carry out the studies. thanks to the novel methods, the time to bring a new drug to market is reducing because in silico computational models are available where you can screen thousands of compounds today in no time. But nonetheless, if you look at this, it takes approximately 1.5 years. So by the time you reach phase 1 or preclinical, almost 4.5 years have already elapsed. And from a set of 10,000 compounds, you have narrowed down to only 10 to 20 candidates, right? And from this 10 to 20 candidates that you have submitted for further development, including phase 1, 2, and 3, only one goes to the market at the end of the day. And this entire scheme of thing requires approximately $2.5 billion of investment, including opportunity and sunk cost. Right? So the companies will need to be extra careful when they generate data. And the failure at this stage to correctly identify the toxicity profile of a drug will eventually lead them to expend this money and get the drug failed at phase 3, which is even more economically unviable. So companies will need to ensure that the toxicity data, as Dr. Sebastian mentioned, needs to be based on complete science, total science, discuss, and make them understand why you're carrying a particular type of study in order to get the approval, and then continue further. Otherwise, the failure in phase 2 and phase 3 is very expensive for the companies. So the GLP requirements in pharmaceutical industry are extremely important because this data becomes, as we know, that IBT case has given us a kind of understanding that if you do not have a strong data which is reproducible and which is acceptable by the regulatory agencies, you are going to fail at the later stage. Now, there have been requirements, there have been changes in Schedule Y of GMP, which has been again replaced, but this is the requirement by Indian regulators. Now, I'm not going into details of whether this is correct or not correct, because these are all evolving things, right? Science evolves and so does regulation, but we are talking of what they are demanding as of now. Now, one of the things that I've noticed is that all those requirements are for small molecules. And when we talk of GLP requirements, we are just talking of small molecules. This requirements doesn't account for some of these newer therapies that are emerging. Large molecules, biotherapeutics, biological molecules, which are not easy to test, which are not easy to manufacture, which are not easy to produce. We do not know what the regulations mean because, again, the regulation is silent on what is required. A very convenient case by case is what is used in the regulation, right? No, they are coming through proper regulatory studies, but there are no stringent requirements. It's not that, okay, A, B, C, D, E is must. So a sponsor can say that we are carrying out this study or we're excluding this study for so and so reason, and based on which they are developing the drugs. So a lot of requirements still exist, but some of those requirements which are not mandated by regulation are also being performed by companies, so as to be on safer side, right? There are, but since the cell therapy itself is in a growing stage, we do not know what regulations exactly has to be followed. Some basic template is available, but that's not a stringent guideline like Drugs and Cosmetics Act, right? So for biologicals, cell therapies, even today you can look at devices, we don't know how the devices have to be tested on humans, what are the requirements. So the regulations are evolving. And we are a little far behind in terms of other agencies as far as regulations are concerned. But hopefully we should be able to address all these regulations going forward, and it should be a part of mainstream discussion. If you're looking at the requirements for regulatory agencies, all this should be a part of your mainstream discussion, which unfortunately is not happening at the moment. NGCMA, sir? Yeah. Yeah, that is NGCMA, National Good Laboratory Practice Compliance Monitoring Authority, which is a part of DST. Sir, DST. DST accredits. So they have, sir is one of the inspectors, so they have accredited, okay. So they have inspectors who visit these facilities and submit the report, and based on the requirements that are listed out, according to GLP requirements. Quality Council of India supports in training this particular. Quality Council of India is a facilitator. Correct me if I'm not wrong, sir. Training programs, yeah.
Also the inspection.
Yeah.
The logistics are
Done by
supported
QCI.
by QCI. By QCI.
The
They are
science is
trained, they
taken
train
care of
in
by,
other things.
yeah. The science is taken care of by DST, logistics is taken care of by QCI.
They have, you know,
It is already involved. It is involved, not in science, but in arranging science, or managing science rather. Okay, so I'll skip some of the slides. These are the requirements that I've taken from Schedule Y 2019 amendment, and many of these documents that are required, many of these kind of studies that are mandated have been discussed by Dr. Joseph earlier, right? So these are the studies that are required, but if you look at some of the studies, I'll just, sorry. These are single dose toxicity studies, and this is a fixed, what do you say, formula which is given. Now, whether this is right, as Sir was mentioning earlier, whether this is right, wrong, species, number of animals, is again, I don't know whether it's based on discussion, science, but they have already prepared this, and it's mandatory for pharmaceutical companies to follow. Now, again, this is where Dr. Joseph's intervention comes, that this needs to be discussed, right? Regulators cannot simply lay down the guidelines without having a discussion, and that's where people who have experience in industry, experience on working in labs, should be involved in making these decisions, right? So thankfully, some of these changes are happening on ground, but the impact will be felt at a later stage. I mean, you may not see the impact in short term, but over a period of time, because this entire system needs to be overhauled, so it's going to take some time in order to see those impact on ground. So GLP basically is a smart approach, and this is my own creation probably, and with this probably I'll try to make a sense of what I've spoken and put all these pieces together. So in order for compounds which are to be used after regulatory approval, one needs to have a smart approach. It includes pharmaceutical also. So your systems should be based on scientific parameters. It should be stringent. One needs to be methodical in terms of approach, and that's where GLP talks about a lot of things, including personal, test system, facilities, documents, quality, control, audit, etc., etc. So everything that you do has to be systematically documented. It should be methodical. The data that you generate should be accurate, and it should be auditable. Accurate in terms of, as I mentioned in case of IBT, they did not document the replacement of animals, right? So it's important that whatever minor changes happen in the protocol should also be documented so that you can justify at a later stage when you go to regulatory authorities for approval. Reliable, reproducible, and robust. So your data, if somebody uses, should be reproducible. And that's where we have all these methodological approaches that requires that today the scientific integrity requires that whatever data is presented should be reproducible by anyone who is interested to work on that. And your test facilities should be transparent in terms of generating this data and submission to regulatory authority. So if we use the smart GLP approach, I think most of the things that we have discussed can be taken care of. Some of the challenges, of course, any good practice requires a truckload of documents. So document is again a challenge, and we always crib about all this documentation when we talk of good practices, but it's nonetheless a part of it, because we know that only with the documentation you can unearth some of these unscientific events that probably would have taken place. It is cost intensive, but as I mentioned, if you invest certain cost, it's possible for you to recover. So in fact, it becomes an investment rather than a cost. But normally what happens when we look at this investment, we look at short-term gains rather than long-term benefits. Emerging technologies and its integration with GLP needs to be discussed. I mean, there are a lot of emerging technologies. Today, people use artificial intelligence, right? How do you use artificial intelligence in animal toxicology? How do you generate that data? Whether that data is robust, whether that data would be acceptable by regulatory agency, we do not know. But companies have already started using this particular technologies, right? As we discussed, large molecules, cell therapies are coming into market, but we do not know how they have to be integrated with GLP or good laboratory practices. There are new therapy areas which we need to again discuss. Medical devices is one area where I see a lot of scope in terms of adhering to medical good laboratory practices. How is a matter of discussion and debate, but there are a lot of areas where we can expand the scope of GLP implementation from the current scope that we have discussed. Right, so this is the summary of what I've spoken. The history is always replete with the context where the tragedies which unfold makes us human realize that we need to have certain good practices. Those good practices, if implemented, can also have its indirect impact in terms of creating a kind of ripple effect, thereby contributing to the nation and pharmaceutical industry to which this is required. If they follow GLP guidelines, our Indian companies are not into the scheme as of now in terms of new drug discovery and development, but if they are trained properly, if we mandate these facilities, again, it could be a game changer for the sector that we are talking of, right? So that's the summary of what I've spoken. And as Dr. Sebastian came, I spoke, but I'm also here to learn, and it has been a good learning experience as well during the previous discussion. And I'm open to learning through your comments, questions, if you have any. Thank you so much once again for the opportunity. I've skipped a lot of slides for want of time, but they're all data that is available. So when I was preparing for GLP, I thought there is so much, yet there is nothing, because the data is available. But how you use that data in order to understand, implement, and use GLP principles for the betterment of humanity and science is something that we need to discuss. And in a way, it's a tribute to Sir, because he has been a pioneer in terms of what he has brought in terms of GLP to our country. So a kind of tribute to Sir as well for the efforts that he has undertaken in this 20-25 years for making GLP a part of discussion. Thank you so much. If there are no questions, at least comments for my improvement. I don't know if something that I've missed out, something that you would want to add. Okay, fine. Yeah. That's where the challenge is, right? So when I say discussion with stakeholders, it's not about putting a draft on the website, inviting them and having a discussion. Yeah, that's true. True. Absolutely. Beginning, okay. Okay. True. 2019, right. And as far as these requirements are concerned, as you rightly said, again, if you all go to NGCMA website, there is a comment section. I mean, they are asking all the stakeholders to provide the requirements of what tests should be included as a part of GLP. There's an Excel sheet where you can already identify and comment as to whether this study should be or should not be a part of GLP requirements for IND submission as far as regulations are concerned. So that again is already available. So if somebody is interested, can go and look at that document and submit your comments. But that's again, it's a valid point, because many times we, from our perspective, often see this drafts being uploaded on website after the due date of comment is there, right? So it's one way of making it available, but I think it's one-on-one discussion with the important key stakeholders is again important. So that's my perspective. Yeah, yeah.
Narrowed down to what is the requirement. We can simply refer and adopt that. That's the very easiest thing. Comments, discussions, all happened at various levels internationally, deliberated using all historical data, thorough analysis done. There's nothing for us to reinvent actually. It's much easy, kind of adopt the ICH, international guidelines, what the other matured,
countries are doing.
Yeah, so So that's the easiest way, instead of we deliberate, discuss, and then narrow down.
No, it's important to adapt rather than copy it, because our context may differ, but it's a good starting point for all of us. Yeah.
The physiology of a human body and animal,
Will
it
remain
remains
same.
same, whether it's India or US. So, that way the science, I think, more or less the same. I'm talking about the data requirements, from a safety evaluation point of view. But other requirements you can set, taking our context into account. But the data requirements, safety data requirements, I think it remains same, wherever you go.
True. Yeah, ma'am.
And it's the same number in phase 1 as well, which is designed to address safety.
True.
So what is the take home from this, just looking at this broad figure? Do you think that the phase 1 safety, therefore, is something that reached phase 3 has actually passed phase 1 safety? Yet it fails in phase 3 because of the numbers, the diversity of individuals who are being tested.
Absolutely.
So then is our phase 1 safety design actually, the recommended design actually the correct design? Because basically, it's the same failure rate in phase 1 and phase 3. Need we look at it? Need we establish the fact, you have SIP diversity in India. You have so much diversity in terms of the, the population diversity, human population diversity in itself. Should Indian designs be therefore catered to have that diversity incorporated in the phase 1 design itself? Is there any discussion on this aspect?
Discussion is not there, but as you rightly said, this safety, sometimes pharmaceutical companies are aware of some of this toxic effects, but considering the risk-benefit ratio, they probably go ahead with phase 2. And when they experiment on large population, that's where they realize that, yes, this data would be problematic going ahead with submitting to regulatory agency, and that's where they drop. Right? So, and many a times if you see some of this data available, this is internal data which is not submitted sometimes to regulatory authorities as well. Right? And that's why a lot of drug recalls happen when they go to market and pharmacovigilance program is implemented. Yes, sir. Yes, sir. Sir, yeah.
How far and wide and widespread is falsification of GLP records and contract?
Sir.
And GLP certification has been withdrawn in India from 2 organizations which I know of. So that kind of thing will happen because inspectors will be going only and do the sample check. Unless an ethics come, that is
Yes. That's why, yeah, sure. Yeah, so, correct.
Scientists, most of the time, they think that ethics is something external. Ethics is not required for science. So that is the biggest crux.
Yes.
That is what we have
True.
felt in ethics committees.
So that comes from within. So now nobody can
has to
teach
come from
ethics.
within.
It is from within. You can have hundreds of training programs on ethics, but if it doesn't come from within, it has no sense.
But training programs have to be there.
You can
cannot
train the
say
people, but if person from within doesn't want to change, any number of training programs will not work.
As you are having GLP, the clinical has GCP.
Yes.
We are having GCP. It is very
Sir,
becoming
in
very
any
difficult
good
to
practice,
implement GCP.
there is a falsification of data that happens. The only thing is
Plagiarism,
that we,
falsification,
absolutely.
fabrication,
That's all
these
scientific
things are
misconduct that happens. But as Sir said, and I understand that this GLP's facilities are, they have to have renewal every 3 years.
Unless-
And there is a surveillance audit also that happens, which is a surprise audit for them
Unless-
to understand.
Unless people who are doing GLP studies, they realize that
Yes.
ethics is a must. They have
The buck stops with us.
buck stops
Yes,
with
yes.
people,
Yes.
with the scientists.
Yes, sir. Thank you, sir. Thank you. Thank you.
Thank you, Dr. Janoria. That encouraged a lot of discussion, and there was a lot of learning as well. Thank you so much for initiating that. And as I mentioned previously, also, we are extremely blessed that we have a galaxy of people joining us today. We have been talking about the GLP practices and NGCMA. Who better to deliver the next talk than the head of NGCMA herself. So we are joined online by Dr. Ekta Kapoor. She presently heads the National GLP Compliance Monitoring Authority of the DST, Department of Science and Technology. She has an experience of 2 decades in GLP certification. The committed and sustained efforts of Dr. Kapoor have not only led to India becoming a full adherent to mutual acceptance of data in the OECD, but also to India's presidency in the OECD's working party on GLP for the next 2 years, that is 2024 and 2025. India will be heading the presidency of this organization. Dr. Kapoor also heads the Frontier and Futuristic Technologies Division in the Department of Science and Technology. She leads DST's 2 national missions, the National Mission on Interdisciplinary Cyber Physical Systems and the National Quantum Mission. So we are extremely delighted to have you as our speaker today, ma'am. So she will be talking about India's journey towards full adherence to mutual acceptance of data. Over to Dr. Ekta Kapoor, please.
Thank you so much. I hope I am audible. Okay,
Please go
great. Respected Director Ma'am, CDRI Lucknow, respected senior faculty members, and my co-speakers in this, short program that has been organized by CDRI. It's a privilege to, get a chance to speak to all of you and tell you about the National GLP Compliance Monitoring Authority, how we have evolved from a non-OECD member to becoming a full adherent to mutual acceptance of data, and how we have further climbed the ladder for, having the presidency of the highest global organization, that is OECD, for the purpose of good laboratory practice. This surely, tells the commitment that India has always had with respect to the quality system of good laboratory practice, and that it also says that how we further plan our journey to becoming sustained leaders in the good laboratory practice certification and compliance monitoring, so that the regulators all over the world are rest assured the data emerging from the Indian GLP certified test facilities is reliable and accurate. So may I just request the presentation that I sent to be shared. And in the meantime, I would again like to thank the organizers, thank Director Ma'am for her kind invite. I'm extremely sorry I could not join physically because of some other pressing engagements and immediate meetings which had to be attended to. So I just start my presentation with telling you some, next slide please, some background on how is India placed with respect to various types of chemicals. So we all know the country has rightly been called the pharmacy of the world. The Indian pharmaceutical industry is third in the world for production by volume of medicines, and 14th position it has overall in terms of value. As of 2021, India was the largest producer of low-cost generic drugs, which is advantageous for exporting medicines to other countries. And the global share of pharma export in 2021 was approximately 20%. Next slide, please. Further, moving to the vaccine production, we have all witnessed the success story of the COVID period, where India emerged as a key player to combat the pandemic, worldwide. And India also, after having more than 200 crores of vaccines in the country, India supplied over 201 million doses to 100 countries across Southeast Asia, South America, Europe, Africa, and the Middle East. So this growth of vaccine production again in the country is of critical importance. It is not only for the benefit of the healthcare of Indian system, but also for the global community. Next slide, please. Coming to the agrochemicals, India is the 4th largest producer of agrochemical in the world and 13th largest exporter. The agrochemicals market, all over the world, again, India has a great stake, and we further look forward to increasing the CAGR of a growth rate of 6%, which has happened in the last year, we further think of exceeding the growth rate by more, beyond 6%. That is the last growth rate. Next slide, please. So when we come to the robust testing programs or systems that are available for various types of chemicals in the country, we start from the idea or hypothesis development, where we think that innovations are the future, and we go about making generating a new chemical entity. When this new chemical entity is generated in the lab by synthesis processes of advanced chemistry, then we conduct experiments in live organisms or animals, where we call them as preclinical studies. These preclinical studies also include the in vitro experiments. Further, while the development of the pharmaceutical products, if I just stick to one example, the safety and efficacy testing is done, where strict GLP guidelines have to be followed, and data which is generated, it has to be robust and reproducible. Further, when the preclinical testing and safety efficacy testing has been completed, the compound or the new chemical entity is subjected to human testing through clinical trials, where a good pool of human subjects and different diseases are tested upon by the NCE. And finally, the process of regulatory filing is done, where the data as a dossier is submitted to Indian and foreign regulatory agencies for approvals. So the various categories of products which are covered under GLP for such type of testing in the country include industrial chemicals, human pharmaceuticals, including biotechnology products, including cell and gene therapy products, vaccines, both human and animal, veterinary pharmaceuticals, medical devices, food additives, feed additives, agrochemicals, and cosmetic products. Next slide, please. So we can say that the pharma ecosystem and infrastructure in the country, is quite strong, with the highest number of US FDA compliant plants being situated in India outside USA, and more than 3,000 pharma companies are there in India, with a network of 10,500 manufacturing facilities. Again, manufacturing facilities in the country have expertise in low cost generic patented drugs, as well as end-to-end manufacturing of bulk drugs and formulations. We have highly skilled resource pool of chemists and formulation development experts. And there is, continuous proactive efforts being carried out by the government of India, by Department of Pharmaceuticals, where launch of the largest national health protection schemes has been carried out. Next slide, please. So whatever I have said till now, I will just like to sum up by saying that as a country, we do understand the significance of mutual acceptance of data systems. The drugs and chemicals developed and exported in the country are of high quality and are safer to use. India can definitely be a future destination for many testing programs in drug discovery and development of other types of chemicals like pesticides, industrial chemicals, etc. To cater to the continued needs of the export of drugs and medical devices, there is a high need for more contract research organizations to be set up in the country. India is definitely committed to maintain a position of leadership, to continue excelling and thriving through the creation of talent pool, and implementation and compliance to the OECD principles are of paramount importance in the push towards excellence in the conduct of preclinical studies. Next slide, please. So this was just a background. I just wanted to tell you the strengths of the country when we come to conducting preclinical studies. Why preclinical studies? Because the quality system of good laboratory practice deals with the organizational process and conditions under which these preclinical or non-clinical health and environmental studies are planned, performed, monitored, recorded, archived, and reported. Each word in this definition, I must say, has its own importance. And therefore, when we say the studies are planned, there has to be a written document called the study plan, duly approved by the study director. It should include all details of the study, how the study will be conducted, what kind of test systems will be used, what will be the dosage levels, how the analysis would be conducted, and so on and so forth. The study is definitely performed only and only in accordance with the written down study plan and with the written down standard operating procedures applicable to the particular test facility. The study is monitored at each part, at each phase, including the live phases, by a quality assurance unit. Without a QA, the study cannot be called a GLP study. The study results are recorded in raw data formats, which are duly approved by the test facility management. The study results, the study plan, the study specimens, everything related to a particular study are archived for reconstructability of the study at a later date. And the study is again reported in the form of a written down report signed by the test facility management, the study director, and also to include a quality assurance statement in the final report saying that the QA has monitored the study throughout its conduct and the study results can be relied upon. Next slide, please. The benefits of GLP include that it limits wastage of resources, avoids duplication of tests, reduces number of animals used in preclinical testing. It ensures high quality of results and ensures comparability and reconstructability of results. Further, GLP helps to promote mutual recognition of results, thus helping to remove technical barriers to trade. So what we are trying to look at, the benefits, they are multifold. We are limiting the wastage of resources by avoiding duplication of tests, saving time for getting approval of the new types of chemicals, reducing the number of animals used for preclinical testing, and of course helping us to export more and more chemicals by ensuring that they are safe to use to other countries, thus creating an economic benefit for the country. Next slide, please. So when we again talk about MAD adherence, MAD means mutual acceptance of data. It is a multilateral agreement which allows participating countries to share the results of various non-clinical tests done on chemicals. The MAD framework of the OECD is governed by 3 OECD Council decisions, namely the C81, C89, 87, and C97-114 decisions. The MAD agreement is based on the simple concept of tested once and accepted for assessment everywhere. I think it is the only way where we agree that the test has been conducted under GLP means that it has to be accepted by all the countries' regulators, which are also signatory to MAD agreement. It thus helps in collaboration with multiple economies and exchange of data by mutual recognition. It helps reliable data on CLP products to be generated for benefiting the benefiting at global level, and data from MAD adherent countries are accepted by regulatory authorities worldwide. Next slide, please. Just taking you quickly through the history of GLP in India, the exposure started in the country to GLP way back in the year 1991. At that time, India did not have any GLP compliance monitoring authority. Indian manufacturers were dependent on foreign facilities for generating non-clinical data. This was a huge foreign exchange burden for the country. Non-GLP data from Indian test facilities was not accepted by regulators, which was an important technical barrier for global acceptance. Indian test facilities thus approached the GLP compliance monitoring authorities of other countries like the Netherlands, Switzerland, Germany, Belgium for GLP certification. And soon the Indian government and the test facilities in the country recognized the need to set up a national GLP compliance monitoring authority. In short, we call it the NGCMA. Next slide, please. The NGCMA was thus born in August in 2002 with the approval of the Union Cabinet. It was set up under the administrative control of DST with the objectives to operate in accordance with the OECD Council norms, to grant GLP certification to test facilities by examining their procedures and practices, and to provide a network of GLP certified labs in the country and assure regulatory authorities that the safety data received from GLP certified labs can be relied upon. This led to transformation of the culture of generation of non-clinical data and enabled the Indian test facilities to meet the requirements of global non-clinical data as per OECD principles of GLP. Next slide, please. Thus, after the NGCMA being created, India became a provisional member of the OECD's Working Party on GLP in 2003. At that time, since the monitoring authority was set up, NGCMA was involved in setting up its own documentation and implementation of GLP compliance procedures. It was engaged in training of GLP inspectors who could inspect the test facilities when they applied. It was also engaged in sensitizing the test facilities so that we could invite GLP applications and thus certify our test facilities based on our own documentation and procedures. We were also involved at that time in training of test facility personnel and liaising with the test facility management for support at various levels. The first GLP certificate, which was a major landmark and beginning of the journey of GLP certification in the country, was in 2004. Next slide, please. The process further, was further carried on with India attending all meetings of the OECD's working group, now called the Working Party on GLP, as an observer. We were not a full member. We did not have voting rights that time because we were not full adherent to mutual acceptance of data. The provisional member status of India was extended every 3 years till 2009. We also organized various training courses in India with faculty from the OECD working group. These were in the years 2003, 2005, 2010. So we also attended various OECD training courses for inspectors, which were conducted in other countries in 2006 and 2009. And we also witnessed on-site GLP inspections in other countries by the Indian inspectors. I think there were, separate teams which were sent to 4 different countries: the UK, Netherlands, Belgium, France, and Switzerland in 2007 and 2008. This was an exercise which gave us a practical experience of attending GLP inspections being done by other OECD member countries, so that we could get a flavor of how inspections are conducted, how the compliance procedures are met with, and so on and so forth. So this was a great training for the Indian GLP inspectors and for the NGCMA staff as well. I attended majority of these, at least in the 3 countries, UK, Netherlands, and Belgium. And this helped me to get a practical hands-on experience on how to conduct GLP inspections. Next slide, please. So further, our journey towards MAD was to be culminated with conduct of a mutual joint visit or on-site evaluation of the Indian GLP program by the OECD's working group on GLP. The first MJV was conducted in February 2008, where we had some major observations received from the MJV team, and this needed to be further improved. Thereafter, a follow-up evaluation of NGCMA was conducted in February 2010. Again, the team which was evaluating the program did a 5-day on-site evaluation, observing the Indian inspectors how we conducted a GLP inspection, as well as detailed review of all the procedural documents of NGCMA. The report of the evaluation was discussed in detail in the working group, and the evaluation team had several suggestions which were accepted and incorporated by NGCMA. Next slide, please. Thereafter came the, India's key milestone in GLP. This was on 3rd of March 2011, when the country was conferred with a full adherence status to mutual acceptance of data. With this, the country became the third key emerging economy after South Africa and Singapore to join the OECD system for mutual acceptance of data, ensuring the results of non-clinical chemical safety testing done in the Indian GLP certified labs to be accepted in all the participating countries. Next slide, please. So I must say that the road that we took towards MAD was full of pitfalls and great learning experiences. We had all the stakeholders, the partners who helped us in attaining this status. These included the test facilities. Next slide, please. These included our own GLP inspectors, the Indian regulatory authorities, stakeholder departments of Government of India, which are all members of our apex body, which is chaired by Secretary DST, and secretaries of all other stakeholder departments are members. We had the OECD working party as itself to provide us necessary support in terms of mentorship. I also already mentioned the on-site inspections that we witnessed in different countries. So the working party members were very keen on India to get a full adherent to MAD status, because we had so many contract research organizations, we had so many good test facilities which could conduct studies which were being submitted to countries all over the globe. Next slide, please. So after we became a full adherent to MAD, we kept on attending all meetings of the working party and contributing to the actions or to the activities of the working party as members, full members. This we also attended various OECD training courses for GLP inspectors. I think 2011 the training was in Israel. Dr. Sharat Sharma from CDRI was also, a participant in this training along with myself. In 2013, again, we had a training in Japan. 2019, we attended a training in South Africa. India also got a chance to host one of the OECD training course in India in Hyderabad in the year 2015, and there were around 30 GLP inspectors from all over the country, which participated in this training course as a part of other participants from the OECD member countries. This was a huge success, and a letter of appreciation was also extended to India by the OECD for conducting this training course. India has also, contributed towards conducting the on-site evaluations of other countries' monitoring program. So this was a great learning experience again, where I was nominated to conduct the evaluation of GLP programs of most developed economies like Canada, Japan, and Thailand. And this further improved our confidence and the confidence the OECD working group had in India about the quality system being implemented in the country. Next slide, please. India has also been a member of drafting groups of various OECD advisory and consensus documents and has been contributing to the development of these documents. India has been a member of the steering group to develop a best practice guide for training of evaluators for on-site evaluations. NGCMA has also been conducting joint GLP inspections with regulators like US FDA and US EPA, and study audits at the request of various foreign regulatory authorities. India was designated as the vice chair for the Bureau of OECD's Working Party for 21 to 23, and is now designated as the chair for the Bureau for the years 2024 and 25. I think this is a matter of great honor and pride for the country that we emerged as non-members of the OECD, becoming provisional adherents to MAD, then full adherents to MAD, and now India will be leading a group of 58 member economies in the OECD, with the other developed countries sitting, India will be at par with other developed countries of the world. The on-site evaluations of the GLP programs happens every 10 years, and this was completed for India again It was earlier held in 2010. It was due in 2020 due to the pandemic. This was successfully completed in 2022. And because of this, India is now a full adherent till 2030. Next slide, please. Telling you also briefly about the structure, functions, and procedures of the Indian GLP program. Next slide. The slide shows you the organizational chart, functional chart of NGCMA. The highest level body we have is called the GLP authority, or the apex body, which is chaired by the Secretary, Department of Science and Technology, and it has the various secretaries to the Government of India as its members. And then we have a technical committee on GLP, which is a recommending body, which recommends whether or not a certificate should be awarded to a test facility based on the inspection report and the action taken report of the test facility. We have a documentation committee, which is a subcommittee of the technical committee. This documentation committee helps us to formulate our own procedures and have them as written down documents. We have a complaints, grievances, and appeals committee. An NGCMA secretariat is set up in DST. And we also have a pool of trained GLP inspectors who conduct trainings for all of us, for trainings, and as well as conduct the GLP inspections of the test facilities. We have global linkages, the OECD Working Party. India is going to chair the Working Party next month. And we are also having a pool of around 60 GLP certified test facilities pan-India. Next slide, please. Also to mention that we have 5 facilities of these 60 certified test facilities. 5 of them are from the government sector, and CDRI is one of those 5. Coming to the scope of the Indian GLP program, we have the various types of chemicals or test systems. I already mentioned all of them in the previous slide. However, the areas of expertise for which GLP certificate is awarded is for these 9 areas that have been mentioned in the OECD principles themselves, and they cover physical chemical testing, toxicity studies, mutagenicity studies, environmental tox studies on aquatic and terrestrial organisms, studies on behavior in water, soil, and air, residue studies, studies on effects on mesocosms and natural ecosystem, analytical and clinical chemistry testing, and under others, we may have areas like safety pharmacology, drug metabolism, pharmacokinetics, etc. Next slide, please. The GLP certificate, which is issued by NGCMA, is bilingual, both in Hindi as well as English, and it looks somewhat like this, mentioning in detail the areas of expertise, the types of test systems, and the types of test items. Next slide, please. So as I already mentioned, by the end of 2023, we have around 60 GLP certified test facilities in the country. These belong to test facilities which are testing only agrochemicals. They may be manufacturers of agrochemicals themselves, or they may be manufacturers of pharmaceuticals, which may be testing their own products. And we also have a number of facilities which are contract research organizations, which conduct studies for the products manufactured by other manufacturing companies. Next slide, please. The Indian GLP program salient features are that the program is voluntary in nature. Certificate is valid for a period of 3 years. The certified test facilities are subjected to mid-cycle surveillance. There exists a procedure for appeal by the test facilities. Procedure is also in place for conducting surprise inspections by NGCMA, and NGCMA reports any information on non-compliant study or test facility to the OECD Working Party on GLP. It is mandatory for us to report all such information so that the information can be percolated to all the members of the working group, so that the data which is being generated is very transparent and it is in compliance with GLP. Next, please. The procedural document of NGCMA, again, these are our SOPs of the monitoring authority. These have to be in compliance with the OECD principles of GLP. They are drafted by a documentation committee, implemented through a documentation control system. And currently there are 21 procedural documents which cover all the procedures with respect to GLP compliance monitoring and administration of the GLP program. Next slide, please. So we have trained GLP inspectors to conduct GLP inspections for us. We have one full-time inspector in NGCMA, that is myself. We have part-time inspectors, which are only and only from the government or public funded institutions, R&D labs, and government universities. We have 16 inspectors which work as lead inspectors or fellow inspectors. And the evaluation of GLP inspectors is a continuous exercise which goes on during the training, during their observerships, by the feedback we receive from the test facilities. And we also do continuous capacity building of the inspectors in critical areas through refresher trainings, which are organized every year. Next slide, please. The slide tells us the various types of GLP inspections that we do in India. The first inspection that is done for any test facility is called the pre-inspection, where we get to know the type of quality system they have put in, the type of studies they are carrying out, so that a final inspection can be done with great emphasis on the studies that need to be audited. So final inspection covers detailed study audits of all the types of studies, at least one study of each type that the test facility has claimed in its GLP compliance. Next is the surveillance inspection, which is midterm. It is conducted 18 months from the date of certification or recertification. Extension in scope inspection can be done for a test facility who wishes to extend its scope of GLP certification post its certification being done. Then after the 3-year period of GLP certification is over, we do a recertification inspection. Surprise inspection can be done with the approval of Secretary DST with a special procedure for test facilities where we receive any complaints or we are not sure of the GLP compliance for various studies that have been submitted to regulators, be it in India, be it or worldwide. Then the verification inspections are done to verify the ATR, the action taken report for a particular inspection by a particular test facility. Of course, under mutual acceptance of data, we are bound and it is mandatory to conduct inspections at request of other compliance monitoring authorities or foreign regulatory authorities, where these regulatory authorities have received data or studies from our GLP certified test facilities. Next slide, please. So just to carry out, to assess the impact that the GLP program of India has had on the country's chemical industry, we conducted an impact analysis study of the GLP program. And according to this, we came out with the tangible benefits of GLP and mutual acceptance of data for the Indian industry. So the test facilities have shown the data and the interviews that we had with various stakeholders. They have shown that the GLP program has had considerable impact on the not only on the conduct of studies, but providing employment opportunities, getting services from various countries across the world, and the conduct of GLP studies worth more than thus contributing to the dream of Honorable Prime Minister of the Atmanirbhar Bharat or self-reliant India. Next slide, please. We do a lot of capacity building activities as a part of our mandate. We do sensitization workshops to invite new, more applications for GLP. We provide trainings on GLP OECD principles for the inspectors, both basic as well as refresher trainings. For test facilities, we do tailor-made training programs. I think each GLP certified test facility keeps on, receiving emails for nominating their participants. We do trainings for study directors, quality assurance personnel. This month only we did one training, 3-day training for the study directors. Participants from CDRI were also, there for the training. We do trainings for IT personnel, for test item custodians, for archivists, and so on. Next slide, please. So we also pay more focus and attention towards harmonization of approaches. These training courses, which I just mentioned, are conducted as per an annual training calendar. The training programs present the basics as well as introduce the updates on the new and revised OECD documents once the documents are declassified. So lectures are always delivered by experienced faculty members. We also have hands-on workshops conducted as a part of the trainings. All test facilities nominate their participants, and all training programs are conducted free of cost. Next slide, please. The major strengths, again, we have a lot of harmonization through discussions and clarifications. As receiving authorities are also represented on our technical committee, there is harmonization and a good working relationship between our own regulatory authorities and the compliance monitoring. Next slide, please. So these are just some clips from the on-site evaluation of the Indian GLP program, which was conducted in July 2023 last year. Next slide, please. I would also like to mention that this is okay. This is the last slide that I now want to present, that the future plans or the way forward for the monitoring authority is that we have already digitized the entire program from a manual to a fully digital mode with respect to our own procedures and processes, including our capacity building activities. So post COVID, we got this great exposure, and I'm fortunate that today I'm able to communicate with you sitting in Delhi only because we are now digitized, and we got an option of online presentations. As a member of the OECD Working Party on GLP, India intends on building a roadmap and evolving the OECD principles and test guidelines, keeping in view the forthcoming challenging areas of risk-based assessments, data integrity, inspection of cloud-based softwares, in silico or QSAR models, and in vitro testing, etc. We also want to handhold with non-OECD member countries and non-MAD adherent countries towards establishing global standards of GLP and aligning their systems with OECD principles. We want to handhold government labs in India to bring them at par with the global standard for the non-clinical research. We also want to expand the scope of Indian GLP program to cover new areas of expertise as applicable and as required by the Indian regulators. Of course, we are committed to implementing a system of GLP certification of highest commitment to quality, which contributes to the dream of Atmanirbhar Bharat or self-reliant India. Thank you so much. And I'm open for any questions that you may have.
Ma'am, thank you very much for that overview of NGCMA and making us understand the functions of NGCMA. If there are any queries, quick comment. Okay, so that's in interest of our time as well. So we'll have to move to the next section now, which is about the reflections and recollections of Dr. Sharad Sharma's journey at CDRI. Since we have assembled here for honoring him for his retirement, so we'll be inviting our former colleagues and present colleagues as well for sharing any of their reflections and recollections with us. So to begin with, may I request Dr. Ekta Kapoor, ma'am, since she's online already. So ma'am, your few comments on your association with Dr. Sharad Sharma, please.
That's, I'm overwhelmed, at this moment with joy and with, there are mixed feelings, but Dr. Sharad Sharma has been a continuous support, not only, in the professional career, as, as a potent, as a scientist, he's also been a great implementer of the good laboratory practice systems in CDRI as a whole. I've seen him, as my own mentor when I entered the DST, and when I joined the National GLP Monitoring Authority. I met him for the first time where he was in the capacity of a GLP inspector. I could observe the skills that he had, honed over past few years. And then I also got a very good chance to travel with him for the OECD training course. And it was great learning always being with him. And we used to discuss at length the, challenges that are faced by the Indian test facilities, challenges that India as a country would face in the OECD scenario. He used to always contribute towards, establishing the standard operating procedures or documents of, the national level, where we used to discuss at length. And it has been, again, it was not very good for me when CDRI got GLP certification. So we had to do away with the services of Dr. Sharad Sharma as a GLP inspector because of a potential conflict of interest. However, while being at the other side of the table also, I found him to be, always engaged in contributing towards doing good science and doing good GLP, both. So my, all the best wishes to Dr. Sharma for always being there, for, helping not only the institute, the CDRI, to grow at its best, during his tenure as the test facility management and the deputy test facility management earlier, but also, helping other organizations also to grow. He's being so kind at heart, always so considerate, always having a helping attitude. I wish him all the very best for all his future endeavors in his, personal journey, as well as if he still wishes to contribute professionally. I think it will be great for CDRI or any other institute where he wishes to contribute. So thank you so much, Dr. Sharma. It was really a very nice period being with you, and I look forward to having more, support in future. Thank you.
This conveying his thank you. Thank you, ma'am.
And thank you so much.
Thank you so much, ma'am. Now we'll open the floor for other attendees. We are joined by multiple former heads of the division. Dr. Nath is saying that he wants to make a comment. Yes, sir. Dr. Chandeshwar Nath, former head of toxicology division.
मित्रों, शरद शर्मा के क्या कहूँ मैं, तो बहुत यादें इनसे साथ जुड़ी हैं और वो तो बहुत समय लग जाएगा। ये जितने बेहतरीन वैज्ञानिक पैथोलॉजिस्ट हैं, उससे भी ज़्यादा ऊंचा बेहतरीन बने इंसान पाया है। हर एक के लिए सहायता के लिए तैयार रहते थे, कभी किसी को ना नहीं कहते। और उसका उदाहरण मैं अपना ही, मैं फार्माकोलॉजी विभाग में जब था, मेरे एक पीएचडी स्टूडेंट को हिस्टोपैथोलॉजी की जरूरत थी, तुरंत इन्होंने पूरी हेल्प की और उसका अच्छा नतीजा। हमने 2-3 रिसर्च पब्लिकेशन भी हम लोग साथ में हुए। अब कहने को तो बहुत यार समय बाद बहुत लोग इच्छुक होंगे बोलने के लिए। मैं सूक्ष्म में इनका एक जो महत्वपूर्ण योगदान शेयर बताना चाहूँगा। रेगुलेटरी एनिमल स्टडीज़ की जो GLP का अभियान सुधीर ने छतर मंजिल में जो शुरू किया था, उसको शरद इनके सहयोगियों ने इस बिल्डिंग में सफलतापूर्वक प्रामाणिकता के गंतव्य तक पहुंचाने का जो कार्य किया, वो एक बहुत महत्वपूर्ण योगदान CDRI को। अब कुछ में अब हफ्ते भर बाद ये हमारे ग्रुप में आ जाएंगे, वहाँ भी इनका स्वागत है और मैं इनके और इनके परिवार को मेरी हार्दिक शुभकामनाएं। और आज हम होली के पृष्ठभूमि में मिल रहे हैं, तो CDRI के परिवार के समस्त सदस्यों को मेरी शुभकामनाएं। धन्यवाद।
Thank you very much, sir. Dr. Rath has provided me a list of those who have actually volunteer to speak. Dr. S.P.S. Gaur, our former head of clinical and experimental medicine.
What can I say today? It's just such a nostalgic moment. I'll take just 30 seconds to speak about me as well, because that will give way to speaking about Sharad. Two of the institutions that influenced my life was the Medical University KGMC and CDRI. लखनऊ मेडिकल यूनिवर्सिटी वो हमारी कार्य क्षेत्र था और ये मेरा कर्म क्षेत्र था. मैंने मेडिकल यूनिवर्सिटी में बहुत कुछ सीखा और यहाँ पर बहुत कुछ मैंने किया और बहुत कुछ पाया. और given a chance, it was a wonderful journey from there to here. Given a chance, I will not take a fraction of a second to relive all the entire moments. And in that journey, probably CDRI, one of the sweetest person that I met is the person sitting there. His smile is such a sweet smile that I don't know, we don't interact in words. We interact more in expressions. हम लोग के भाव इतने मिलते हैं कि शब्दों की ज़रूरत कम पड़ती है. तो नॉन वर्बल नहीं, नॉन वर्बल इज एन अंडरस्टेटमेंट. भाव समझना, मतलब उसमें नॉन वर्बल भी नहीं है. इवन इफ इज नॉट देयर, मुझे कोई चीज़ की ज़रूरत अगर शरद से है तो मुझे इनकी शक्ल भी देखने की ज़रूरत नहीं. मुझे पता है कि ये काम अगर हम शरद से कहेंगे तो शरद कर देंगे इसको. तो वो नॉन वर्बल कम्युनिकेशन से एक स्टेप अलग की ये डायनेमिज्म है ऑफ भाव समझना. तो आई विश हिम ऑल द बेस्ट in future life. Anything that I can do for him, it will be a pleasure doing that. Professionally, everyone knows how good and how wonderful he is there. But as a person, he is probably one of the sweetest person in CDRI that I have met. All the best.
Thank you, sir. Dr. Naibedya Chattopadhyay will be speaking.
वो दिन याद है शरद, हम लोग ओल्ड कैंपस में चाय के लिए जाते थे. एक साथ जाते थे, बीएन वगैरह सब एक साथ हम लोग जाते थे चाय पीने के लिए. इनका जीएलपी का काबिलियत ये सब के बारे में तो बोल ही चुके हैं. हमारे जो कैसी ऑक्सीडेंटल स्टडीज़ किस तरीके से इन्होंने सफलतापूर्वक इसको अंजाम दिया, ये भी जानते हैं हम. लेकिन मैं एक चीज़ बताना चाहता हूँ जो मेरे को लग रहा है कि इनके जाने के लिए जो हम लोगों के दिल में जो एक वैक्यूम है कि अभी वो नहीं रहेंगे, हम लोगों के साथ अपना ज़िंदगी जिएंगे. उसके अलावा एक बैड न्यूज़ है फॉर ऑल साइंटिस्ट लोअर देन जी. वो ये है जो डॉक्टर गौर अभी बताए थे कि हेल्पफुल, नॉट ओनली हेल्पफुल, वेल विशर. इतना स्ट्रॉन्ग वेल विशर है कि वी हैव वर्कड इन द कॉलेजियम एज वेल एज इन ये एम्पावर्ड कमिटी. पूरा होमवर्क करके आते थे ये, और इसलिए होमवर्क करके आते थे कि कोई भी साइंटिस्ट को कितना ग्रेड मिलने से इनको या तो मेरिट हो जाए या तो मोस्ट ऑफ द केसेस में उनको रियल टाइम प्रमोशन मिले. ये टोटल होमवर्क करके आते थे और उसके लिए वो फाइट करते थे कि अगर इवन द कमिटी इज़ नॉट इन फेवर ऑफ गिविंग द grade which he is proposing, but he will continuously fight and make it happen. So that is what I am saying, that for those who are below scientist G, you are going to miss a real well-wisher. This is something which I think I should share with all of you, because I will also miss you very soon in that capacity with the director sitting here. So this is what I think that will be something we have to see how his best wishes can be the tradition which he has left, how that can be implemented. We will say that during the next meeting, we most probably will say that assume that if this grade would have been given in presence of Sharat Sharma. All the best, Sharat. All the best.
Thank you, sir. And I invite Dr. Sudhir Srivastava, former head of toxicology.
If I am to speak about Dr. Sharat Sharma, I'll have to talk about how the toxicology division shaped up during the 90s. Earlier, when the CDRI was working since 1951, it was focusing mainly on drug discovery programs. And we were not getting adequate attention in terms of the development programs. And now, since the 90s, we started getting a due amount of attention. And we incorporated the basic ideas of doing target organ toxicity. The dose range finding toxicity studies were incorporated for the first time. Before that, we were only doing safety evaluation studies. We used to go only up to the 10 times the expected dose and then some fractions thereof. And then we would take a limited number of animals for some limited duration of time, and we will declare the drug as safe. That was all. But later on, it was for us, those who were the stakeholders during the 90s, that is Dr. Sharat Sharma, he joined in 1995, and then Dr. Rath, and subsequently followed by Dr. Smriti and Dr. Sarika. So the group working on systemic toxicity studies, they got the chance to work further and to expand it up to the expected levels in the international regulatory environment. And the scientific part of the studies was really upgraded. In the process, we also got a lot of support to upgrade the department, a lot of funds also, and we moved from manual to automation, automated work in a very big way. During the end of this decade of 90s, we got fascinated with GLP also. And even without the GLP monitoring authority being in place, we started implementing GLP in our own systems. And for that, we had the support again from Dr. Sharat Sharma. In 2 words, I will just like to summarize Dr. Sharat Sharma. His name is Sharat. And Sharad Ritu incorporates later part of the autumn and the beginning of winter. So he's such a cool guy. He got it, he really lived up to the reputation of his name, that is Sharad. And I always admire the way he has coolly managed everything in the past 30 years. Just one thing I would like to comment upon. When we joined CDRI, there were three very big names in drug research, medical scientists. Dr. Dhawan, Dr. Nirmal Sethi, our own head of the department, and Dr. Swaran Nityanand. They retired before 2000. And after 2000, I was the first one to go, and probably he is the last one to go as a medical person from the CDRI group. So congratulations on joining the senior league, and welcome.
Thank you, sir. I request Dr. J.S. Srivastava, please.
मेरे कहने के लिए कुछ बचा नहीं है सब लोग इतना कह चुके हैं शरद के बारे में और शरद से हमारा एसोसिएशन तो बहुत पुराना रहा ही है और मैं तो बस इतना ही कहूँगा कि वेलकम टू आवर ग्रुप एंड बेस्ट ऑफ लक and best wishes for your future. So I think you will definitely be picked up by some institution. I know it. So congratulations for that.
Thank you, sir. Our former colleague, Dr. P.S.R. Murthy, sir, is also here, so he would want to say a few words, please.
The time has gone. Thank you. Well, my memories have always been sweet as far as the Dr. Sharat Sharma is concerned. So I treasure those memories because They are very sweet and very cool. In this sense, I think professionally, when we were working together in the same lab, toxicology department, when there was a lot of talk about GLP implementation, and so some of those very earliest lectures were given by me on GLP, and using DOS managed computer systems, slides. So, and that was the beginning. And later, when the first study, I wanted to do it complete in compliance with the GLP. That was on streptokinase, or biotech product of the ex-Director General CSIR, who was the director of VMTech then. So that was, and I insisted, Dr. Sharma, be my quality assurance manager and put your signature on all my raw data. So he did so with a pleasant smile always on his face, as of now. Then it was a great experience, and he has been following me right from then. Okay, let us do some GLP work. Let us do some GLP, and he did it. And today we have a GLP compliant laboratory because of him. Thank you very much. Allah. I wish you all the best and great health. And as usual, keep smiling and make others smile.
Thank you, sir. Our former head of the department, Dr. Neeraj Sinha, could not join us here as he is traveling to Pune, but he has sent a video for Dr. Sharma. So may I request Arvindji to kindly play that.
Hi, Dr. Sharma. Today marks your day of superannuation, a day to celebrate your accomplished career and the beginning of an exciting new chapter in your life. Dr. Sharma, it's a moment to look back at all you have achieved and to look forward to all that's yet to come. As I heard about your retirement, I couldn't help but reflect on our time together, the experiences we shared, the lessons we learned. I think that retirement is not the end, but a transition, a chance to embrace new opportunities and chase dreams that were once on hold. This new phase is your opportunity to explore, the engage in, I think, hobbies and passions. Perhaps set aside and to enjoy the moment of life that work may have previously consumed. It might have happened due to lack of time or very busy schedule. As you embark on this journey, know that my best wishes are with you. I hope this period is filled with joy, good health, and the freedom to do whatever brings you happiness. And remember, should you ever need me for anything at any time, I will be just a call away. Thank you for the memories, the professionalisms, and the friendship. On this very special day, my best wishes to your wife, Dr. Mandira, Shubham, your son, and Shreya, your daughter, who will now benefited from spending some quality time with you. Although it is a very overwhelming times, but now I am feeling little to say some little Sairana mood. So I will tell you two lines for you. रहेगी सदा यहाँ प्यार की ये दास्तान, अलविदा कहने से पहले याद रखना ये अफसाना. My best wishes and blessings are always with you. Thank you, Dr. Sharma.
Dr. Neeraj Sinha happened to be Dr. Sharma's bridge partner as well, in addition to being his head. So probably that's what made him a bit Shairana as well. So moving on, may I now request Dr. R.K. Singh, our former head, to please say a few words.
मित्रों, मुझे बड़ी खुशी हो रही है डॉक्टर शरद शर्मा साहब के बारे में अपने विचार व्यक्त करने के लिए। जब डॉक्टर शरद शर्मा साहब ने 1995 में ज्वाइन किया था तो हम लोग हेमाटोलॉजी लैब में एक साथ काम करते थे और सब लोग एक ग्रुप था बहुत ही अच्छा, और हम लोग बड़े अच्छे से सब्जेक्ट में भी और पर्सनल लाइफ में भी खूब एंजॉय करते थे। लेकिन एक दिन सबका ही आता है जब ज्वाइन करते हैं कि रिटायर होना ही होता है, इसलिए जो चीज़ हंसी खुशी से सब कुछ हो जाए वो बहुत अच्छा रहता है। और इन्हीं शब्दों के साथ ही मैं शुभकामनाएं देता हूं डॉक्टर शरद शर्मा साहब को, उनके परिवार को, कि बहुत अच्छे से समय बीते और हमेशा एंजॉय करें, खुश रहें, प्रसन्न रहें। धन्यवाद।
Thank you, sir. Amongst our present colleagues, Dr. Amit Mishra wants to say a few words.
देखिए, मैं फार्मासिस्ट हूँ, कंपाउंडर, और कंपाउंडर का डॉक्टर की तरफ जो एटीट्यूड होता है वो भय मिश्रित औ होता है। मैं इससे पहले कुछ डॉक्टरों के साथ पीएचडी कर रहा था, जब मुझे पता चला कि डॉक्टर की बहुत से फ्लेवर्स होते हैं। हर फार्मासिस्ट को हर डॉक्टर से इतना घबराने नहीं चाहिए, क्योंकि अलग अलग, खैर दूसरी बात। लेकिन हमारे इंस्टीट्यूशन के लिए शरद सबसे ज़्यादा वैल्यूएबल मेडिकल साइंटिस्ट में इसलिए हैं क्योंकि वो पैथोलॉजिस्ट हैं। और जैसा उन्होंने खुद कहा कि अ क्लिनिशियन knows nothing but can do anything. A pathologist knows everything but can't do anything.
He gets to know it very late. Often when the samples are collected after
That's of course part of it. That's part of it. But he can't do anything. That works both ways, diagnostics, and that's a different debate altogether. My intention is to express appreciation for a great deal that Sharad taught me. He did his residency in a TB ward, and I have made it my life's business to work on TB. And he is among the rare F1 generation CDRI scientists. Before him, his father was in CDRI, and although I never had the good opportunity to meet him, if you look around, are there any F1 generation CDRI scientists? So we are losing F1, meaning his father was there. So we are losing a unique specimen of scientists. नहीं, ऑब्सोलेट कुछ नहीं होना। आप लोगों ने देखा होगा कि जाते जाते मैं इनको एक पैकेट थमा दिया। वो उस पैकेट में कुछ जेम्स हैडली चेस की किताबें हैं, क्योंकि जब ये पढ़ रहे थे कानपुर में GVSM में, और उनका घर लखनऊ में था, तो ये बस से अप डाउन किया करते थे, और रास्ते में जेम्स हैडली चेस पढ़ते थे, जिससे बहुत खुश थे, क्योंकि जेम्स हैडली चेस में दिमाग नहीं लगाना होता है, मेडिकल स्टूडेंट को वैसे ही बहुत पढ़ाई करनी पड़ती है। तो उनको प्रेरित करने के लिए कि रिटायरमेंट के बाद दिमाग ज़्यादा न लगाएं, मैंने इसको एक सेट दिया है जेम्स हैडली चेस की किताबों का, जिसमें मेरी फेवरेट भी है मिस शम्वे वेव्स ऑफ वॉन्ड, जिसमें जादूगर नहीं है।
Thank you, Dr. Mishra. Like a true pharmacist, he gave a gift to the doctor. Well, so next up, Dr. Sanjay Batra, please.
Okay. As I, on the other day, I said that Sharad, me and Kishore, we both were interviewed on 28th April 1995, and we joined. Kishore and myself joined in June, but Sharad, as Amit pointed out, his father was here, so he could not join earlier. Like he has to, he has a cooling or period of 6 months, permission from the headquarters. So it took some time, so he joined later. Later I came to know that he is one of my cousins from wife's side. So, so it was means after some time we came to know. And Sharad, as rightly pointed out by Navid, Sharad has been a masiha for many of us. He has been fighting for many of us behind the scene. You may not even know it, but Sharad has done it. Even for Group 3 staff, Group 2 staff, he has been kind of a very generous person. And I don't know how many of you know, he is one-man army to get your NPA back. If some of the people who had the opportunity to get that NPA, Sharad was one-man army. He has fought nail and tooth like anything. I've seen him. He had made drafts. He has sat with lawyers. He have done so much of thing. I think all should give him the credit for doing that. And of course, Sharad, as you all must have seen, Sharad does not get angry. I have never seen Sharad foul mouth or getting angry or getting frustrated with anything. He absorbs himself, and as rightly pointed out, he is like Sharad. He is very cool. He keeps things to himself, and if you have to discuss anything, he gives us a very right options. He has helped me. He has also took me to doctor. I had some problem of nerves a couple of years back. He has took me to doctors and for discussion. Although now I understand why it was happening, but yes, things have moved on, and we will of course miss Sharad, and we'll be soon joining him maybe next year. Rath, me, Navedya, and me, Rath, Navedya, Amit, all will be in the same league. So thank you, Sharad, for everything, for making this workplace a beautiful one, and we wish you a very happy retired life. I know you will not be retired. You will be doing something. As a pathologist, you have a lot of options to do. So we thank you for everything.
Thank you so much, Dr. Batra. With the loving and popular person that Dr. Sharad Sharma is, where I have been still receiving some names, but what we are going to do is we still have an official retirement party on 28th. So we'll take some of those there. So last speaker today would be Neeti, who will be saying a few words. And then we will hear from Dr. Sharma also, and we have a short video. And after that, we'll conclude.
Yeah, good afternoon, everyone. As Dr. Batra said that Sharad is very cool, but मैंने इनसे बहुत डांट खाई है. डांट एक पर्सनल नोट पे है. I was not going to the doctor for some persistent problem, and इन्होंने मुझे डांट लगा के मुझे खुद डॉक्टर का अपॉइंटमेंट दिलवाया और खुद मेरा फॉलो अप भी किया. So that is something he keeps a personal touch with people and he follows up. And this he has done for the students also. बहुत सारे students हैं CDRI के जिनको कुछ ना कुछ problem होती है तो we as PIs बोलते हैं अच्छा चलो शरद के पास लेकर चलते हैं. So I think we'll miss you a lot and especially आपने हमारी health का बहुत ध्यान रखा सबकी and हमें doctors से connect करवाया because Lucknow was not my hometown 10 years back, but now I feel I'm a Lucknowite. So thank you for connecting with Lucknow and thank you for being with us. Yeah.
Thanks, Neeti. I would second Neeti on that because whenever any of us, colleagues from within the department, outside the department have any issue, we need a doctor's consultation, we would request Dr. Sharad Sharma, and he would have a batchmate in every specialty. And he would call them and help us with even getting appointments. So thank you for that. So we have a short video made on Dr. Sharad Sharma's journey. So let's take a look.
In which year did you join CDRI and how has been your experience?
After completing my education, I joined CSIR-CDRI on 1st August 1995. And I have been fortunate enough that all my seniors of the division at that time, especially Dr. Sudhir.
How was your initial phase? Who did you get support from in the last year?
As I told you that when I joined, I had only rudimentary knowledge regarding the development of any drug entity. And all my seniors, especially Dr. Sudhir Srivastava, supported me and guided me at every step. How to plan the study, how to conduct the study, and how to report the study. Whatever I learned from him.
Sir, what was your work in the laboratory?
In Division of Toxicology, the mandate is to conduct the regulatory toxicity studies of all the chemical entities and drug entities, phytopharmaceuticals which are developed in CSIR-CDRI. And the regulatory aspect is the most important one when you look into the fact that safety is the most important important for any new drug. So it is just like a gateway to the clinical development.
Sir, how was the cooperation coordination of colleagues in your departments? What would you like to share?
In the Division of Toxicology, we have a culture of team responsibilities and teamwork. And it is only due to their support and any studies conducted. Each and every member of this team, whether from the attendant level to the study director, everybody is equally important and has to take responsibility. And I found that CDRI toxicology division people are always helpful for each other. And to do the effort of all the toxicology division as well as other members of the CDRI, we got accredited for GLP studies from NGCN.
Tell us something about your family.
I've got a daughter and a son. Both are pursuing MBA from Mumbai, one from the Nasi Mundi, another from KJ Somaiya College. And my wife is a professional, she is also a pathologist. She is a professor in a private medical college and has her own lab. Grateful to all my colleagues, past and present, for helping me discharging my responsibilities at CSIR-CDRI. My current colleagues, Dr. Arath, Dr. Amir, Dr. Smriti, Dr. Sarika, Dr. Vivek, Dr. Madhav, Dr. Viren, and I'm also thankful to Dr. Ashish, who has just joined, that they have always helped me in all, supported me in all my responsibilities, conduct of my all responsibilities, in discharging my responsibilities.
बंजारा वक्त के कितने शहरों से गुजरा हूँ देखें, वक्त के इस एक शहर से जाते जाते मुड़ के देख रहा हूँ, सोच रहा हूँ, तुमसे मेरा ये नाता भी टूट रहा है। तुमने मुझको छोड़ा था जिस शहर में जाकर,
वक्त का अब वो शहर भी मुझसे छूट रहा है। मुझको विदा करने आए हैं इस नगरी के सारे वासी, वो सारे दिन जिनके कांधे पर सोती है अब भी तुम्हारी जुल्फ की खुशबू, सारे लम्हे जिनके माथे पर रोशन है अब भी तुम्हारे लम्स का गीत।
And I extend my heartfelt gratitude to all my colleagues in CSIR-CDRI and best wishes to all of them.
Thanks to the production team for this wonderful video. Ravindra ji and Kazim ji are here, and Shreya, they have helped producing this video. Thank you so much, and all the best to Dr. Sharma. So moving on, we will now be presenting small mementos to our speakers. So that will be our last concluding part of this program before Dr. Rath proposes vote of thanks. So may I request Director Ma'am to kindly do the honors and present a short memento. Dr. Sebastian Joseph, please. A small memento for Dr. Manthan Janoria, please. Now may I request Dr. Sharad Sharma to please come over. A small memento for you, sir. May I request Dr. Mandira Sharma also to please join us. Yeah, we are extremely delighted, ma'am, that you are here on this special day. Yeah. Dr. Rath, you also join. Thank you so much. Now may I request Dr. Rath to please propose vote of thanks.
Good afternoon, everybody. It is customary to propose a vote of thanks to everybody. So let me start with thanking Professor Sebastian and Professor Manthan. In a very short notice, they could able to make this event successful. Till yesterday night, we are 11 o'clock, we are in chat in WhatsApp, and I was also talking with Madam because Ekta has to cancel her ticket. So I am also grateful to Ekta to immediately ready to come and send the slides so we could able to see her slides and she could make it in proper time. I'm grateful to Dr. Radha Rangarajan who actually did the coordination, frankly speaking, because Madam, whenever I disturb her, immediately she used to reply with a solution, and Madam did that. So we are very lucky to have Madam here as a drug discovery and development person. As Dr. Sudhir Srivastav told, when we joined, it was focusing on discovery. Now Madam is one who is focusing in both discovery and development, and I am sure under her stewardship we will go ahead, much ahead what we are today. I am grateful to all my seniors with a small call, WhatsApp message, Dr. Sudhir, Dr. Srinath, Dr. Srivastava, Dr. R.K. Singh, Dr. Neeraj Sinha, Dr. Gaur, all of them, even two persons they could not able to make it, Dr. Ghatak and Dr. Asthana, who were actually not well. So I advised them not to come. because to handle them also will be a problem. So I am grateful to you all because in short period you have come, and Dr. Dikshit and Madam Dikshit, both of them yesterday, they were telling that there is a meeting and she has to go back. I told, okay, for a short time you can come. She was here in the morning session and she left after some time because of the meeting. I am grateful to all these GLP personals, my dear colleagues like B.N. Singh and Dr. Sanjay. All they have actually, they have some other engagements, but they left everything and came. And my sincere gratitude to my GLP team members, especially those who are working in on the table. They all are here. They cancelled their leaves and they came, so I'm thankful to you. Students, without you, we are actually nothing. And all of you have come today, and please be there and take your lunch and go. Thank you so much again to all of you. Thank you very much.